Good morning. Thank you for coming after yesterday dinner. So, okay, yeah, okay, good. And this one, I don't know what it is for switching slides? The second one? Yes, probably. Okay, so, yeah, I would like first to thank the organizers for letting us to present our activities. We are at the LBP in Irkirch, and I lead the team for Active Materials and Bioimaging Group on development of probes. And we work on three main research directions, fluorescent molecular probes. So this I will explain a little bit today. Fluorescent nanoparticles based on polymers and lipids for biosensing and bioimaging, notably for detection of biomarkers of cancer and viral diseases. And fluorescent biomaterials, those are bulk materials like coatings for image-guided surgery, for example. So I will focus on the first part, especially on the membranes. Does it switch? Ah, yes. Why biomembranes? This is a frontier of cells. It allows you to delimit the cells, but not only the cells, also practically all organelles are delimited by membranes, except membraneless organelles, of course, but practically any organelles known have the biomembrane. So sensing and imaging those membranes is important to monitor cellular functions. And membrane itself, plasma membrane, has a lot of different functions like barrier, nutrients, and transport, signal transduction, etc. And it has specific organization like lipid rafts organization, etc. So we are particularly interested in solvatochromic dyes. So these are molecules which has push-pull structure, which get in the excited state high dipole moment, and they are stabilized by solvents. Polar solvent is a water up, and then you have a polar solvent which are oil, membranes, etc. And therefore, these molecules actually behave like chameleon. In oil, they are blue, and in their water polar solvent they are red. So there is a red shift with polarity, and this is important for sensing. Throughout the years, we develop a series of probes. So those are delimited here, based on NIRAD, based on fluorine, based on Lordan, etc. So those molecules are actually able to monitor lipid organization. And for example, when you compare ordered disordered phases with high level of cholesterol, low level of cholesterol, saturated versus unsaturated, they shift their emission. And this shift in the emission can be visualized using ratiometric imaging by splitting the channel into blue and red and then building the ratio. And so like this in giant vesicles, for example, we can distinguish ordered disordered phases and see the separate phases. And then in cells, we could, for example, monitor change in their lipid organization when cholesterol extraction. Moreover, we can visualize lipid organization at a nanoscale resolution because originally when we look at those membranes with normal fluorescent microscopy, we didn't see any structures. And so for that, this is, you know, very well. A nanoscopy, for example, we based on single molecule localization microscopy, we need something that blinks, but NIRAD doesn't blink really. And so therefore, we actually use paint approach, which is based on reversible binding and unbinding. And actually, our original probe that we developed, NR12A or NR12A, did not blink. So therefore, we developed an anchor group which has low affinity to membrane that binds reversibly to membrane, so that we can generate on-off switching behavior. So this blinking is not intrinsic blinking of fluorophore, but capacity to bind and unbind to membrane. And therefore, in collaboration with Keksu in Berkeley, we're able to do super resolution paint imaging, but even more advanced technique was later used by Keksu when each individual molecule, the emission was split using prism. So like that, we can obtain spectrum from each molecule. And from that, we could construct a map of the emission maximum. And that describes actually lipid organization at the nanoscale. And further, we applied those solvatochromic probes to study organelles. And for that, so the membrane probes I already showed, their lysosomes using specific groups that can localize chemical groups that can localize them in specific organelles. Lipid droplets, hydrophobic groups, cationic groups for the mitochondria, those reactive groups for ER, and long chain for Golgi. And then when we colocalized with the corresponding marker in imaging, we could see actually a perfect colocalization for membrane, for ER, for the mitochondria, for the for lysosomes, for Golgi, and for lipid droplets. And it's actually, you shouldn't take for granted that these chemical groups can direct a fluorophore to any compartment you want. It's a really specific property of NARAD because it is actually neutral molecule, easy to target. But rhodamine, for instance, you cannot target like this, or Alexa dyes in general. So then what we can do when we target those NARAD dyes to specific compartments, we can actually see that each organelle is characterized by its specific lipid organization, lipid order. So the most apolar are lipid droplets and plasma membranes, and all these organelles has different polarity, so lipid organization. And but moreover, we can follow actually some physiological processes. For example, oxidative stress. By applying hydrogen peroxide, lipids get oxidized, peroxidized, which increases polarity. And so we could observe that certain organelles do change their lipid organization because of lipid peroxidation, while others are relatively stable. So oxidative stress is organelle sensitive, and we can follow with organelle-specific probes. And to go beyond single dyes, we worked on fluorogenic dimers, and it works based on aggregation cost quenching principle, where you know very well that fluorescent at high concentration self-quench because of formation of ash aggregates. And we develop fluorogenic dimers where within the same molecule, the dyes can either aggregate and then under stimulus they turn on. And I will give you one example of those dimers where which is used for imaging membranes with superior resolution. So here we develop a dimer of a cyanine 3 and cyanine 5. In water, it's supposed to be aggregated. So indeed, when you measure it in water, you see a excitonic band of ash aggregate, and then it goes to apolar environment of membrane. The absorption spectrum of individual cyanines recovered and fluorescence rise up. So we develop a paint-based cyanine probe that can go on and off. And you can see this is again in collaboration with Keksu in Berkeley. We showed that these dimers show strong light up on binding to the membrane compared to NARAD, much brighter. And another thing that they diffuse much slower on the membrane. And as molecules diffuse slow on the membrane, you can do a better localization, so you can increase spatial resolution of super resolution imaging, single molecule localization microscopy of membranes. So we could increase more than 3-fold resolution on the imaging of membranes. And then as they are bright, we could accelerate also single molecule recording and imaging. So here it's every 40 seconds we obtain a super result image, and we could see already dynamics at the nanoscale. And the last point I wanted to talk is the problem of those probes that I mentioned, including, for example, our recently developed Membrite. They use just to stain membranes. They are they bind non-covalently, so they can desorb in biological media, for example, like serum under permeabilization conditions or in mixture with other cells. So what we develop is the lipid-directed covalent labeling, where we use the reversible binding to the membrane together with the covalent grafting to the membrane proteins. And this is an example of this probe we call memgraft. When you add it to the cell, it stains nicely membranes like other membrane probes. And when we take sulfacyl-3, so this is a commercial compound which will covalently graft to proteins, it labels very badly because there is no lipid-directed process. And where we show the power of this probe, when you put the cells in serum in the native environment to grow cells, one can see that Membrite, our non-covalent probe, disappears because it gets taken by the serum, while the memgraph probe remains for a long time, for hours. And then we can, for example, when we use this covalent grafting, we can label covalently with two different colors, two different cell populations, then mix them together. And after 5 hours or 29 hours, we can track cells in their color at the membrane level. So we can see membrane contacts, etc. And we can track dynamics of cell-cell interaction, cell migrations, again, at the membrane level. And this is done in serum, which was not possible before with Membrite or any of other probes we developed so far. We can go further and start mixing actually the dyes of two colors to generate color codes. And here, by mixing probes of different colors, we could code individual cell populations in 5 different color codes and then mix them together, and then we could recover green, orange, red, etc. individual all 5 populations. I think we can go further, especially if we develop a third color. So in conclusions, we work on soratochromic dye and fluorogenic dimers, which gave us lipid order probes at the plasma membrane or in organelles. We can follow their physiological processes. And then with dimers, we develop super resolution imaging probes. And those recent covalent probes allows you long-term imaging in native environment. And of course, I would like to thank. Okay, these are commercialized probes. I can provide still for free, especially within the network. But I would like to thank my team, especially people involved, collaborators, of course, and different financial resources. And thank you, FBI, and thank you for your attention.
Okay, good morning, everybody. I would like to thank, of course, the organizer, local organizer, for giving me the opportunity to present the work that we are doing in the team of Yves Mély. So I joined his team like 10 years ago as maître de conférence. And so we are also a team of the Laboratory of Bioimaging and Pathology, so another team of the lab. And I selected 3 topics for you today. One related to the use of lipid binding protein to probe lipid at the surface of cells. Another one is a derived paint approach, derived from DNA paint. And the third one is the use of upcounting nanoparticle for single particle tracking applications. So our team has focused on biomolecular interaction. So we are a biophysics team, but we are a pluridisciplinary team with chemists, biologists, and physicists. And we try to address these problematics using fluorescence techniques. So we have 3 research axes. One of them has been already presented by Ludovic Richert yesterday as part of the tech transfer project. So what we are doing in this research axis is using fluorescence analog to nucleotides in order to probe local environment close to the DNA or RNA to probe some DNA protein interactions. Another research axis is focused on the dynamics of lipids and lipid nanoparticles. So some more info about this soon. And the third axis is dedicated to technological or methodological developments. So the particularity of our team is that we are also hosting part of the PIC-Quest platform, the PIC side of this Quest platform. So we developed some homemade microscopes, so spectral paint, two-photon film, anti-Stokes microscopy, 3D palm-storm single molecule and single molecule UV microscope. And all this setup after development in the more research part of our team are then transferred once in production to the platform, so to be accessible to the community. So today I will focus mainly on this setup and the anti-Stokes microscopy. So one of the first topic I'll show you derived from HIV-1 virus formation. So in fact, if you express in a cell this GAG polyprotein, it will start to assemble at the membrane and start to form viral-like protein. And it's very known in the field that the composition of the lipids of the virus is very different from the host cell, and therefore there was some interest into understanding this process. And Nario and Tomishige and Toshihide Kobayashi, they developed a toolbox of well-validated lipid probes and that were used in this topic. But due to the size of the virus, it's necessary to go below the limit of diffraction, and therefore we use super-resolution microscopy. So some word about the probe. So these probes are derived from domain of some of them from toxin. So it's just part of the toxin, so they are not toxic, and they are able to bind to recognize very specifically. For example, here for this entelise, it recognizes specifically sphingomyelin, as you can see by comparing these two images. So this is, let's say, a control experiment, and here we remove all the sphingomyelin, and you see no more signal. So, and there is many probes such as this one. For example, D4. This one binds specifically to coal-rich domain, and using this probe, then we can try to explore the composition of the membrane and its structure. So in this project, what they did is that they performed super localization microscopy. So in D-storm, in this context, with Alexa 647 bound to entelise that recognize SM clusters, and so here you have only the vector. This is a GAG wild type, and this is a mutant of GAG that cannot multimerize and then do not induce the formation of these viral-like particles. So from this diagram, that we could see that GAG wild type induce an increase in the size of the sphingomyelin-rich domain. So this was done by a cluster analysis. So we could confirm this observation and did some other study using fluorescence lifetime imaging. So in this one, we use D4 lipid binding proteins that recognize cholesterol together with entelise that recognize sphingomyelin. And we could observe that in presence of GAG wild type, we had an increase of the Fred population, meaning that we have a shortening of the distance between cholesterol and sphingomyelin, and we conclude that in fact a GAG is able to coalesce pre-existing sphingomyelin-rich domain and GAG-rich domain during the formation of the virus. So I will jump to another topic that is related to paints that was already discussed by Andrey. So in D-storm, you need to have your dye blink. So you have to transfer it from one bright state to some kind of dark state. So in storm, it's kind of random because you exploit the blinking of your dye, but in DNA paint, what is interesting is that you can more or less control this blinking by dividing your system in two parts. You have the docking strand, the imaging strand, and DNA paint allows you also to go a little bit behind because you can increase the final resolution because you can image many binding events. The only problem is that you have some background noise that comes from the floating imaging strands. So one solution to go further is a Fred paint approach, but still you will have a lot of background signal coming from the imaging strand. because if you want to accumulate sufficient amount of signals, then the idea is to increase the concentration of imaging strand in order to increase the rate of formation of the bright duplex. So one solution to go around the still some drawback is to use a quenched dark donor. So for this, this has been done in collaboration with the team of Alain Burger in Nice. So, and the idea is to use this dye, DFK, that acts as a dark donor. So, in fact, it's a nucleobase-based substitute that make it very easy to use in such kind of DNA paint approach. And by excitation, in fact, you have no donor emission in single strand. If you put it in double strands, then you can recover like 5% of the quantum yield. Of course, if you use a complementary strand with acceptor, due to the long Stokes shift for this DFK dye, you have nearly no acceptor emission, which is good for the fret approach. And but if you prepare a duplex with this dark donor and bright acceptor, you can still recover the signal. And what is very interesting in this in their approach is the very large apparent Stokes shift that they obtain using this DFK dye. So in the dark resonance energy transfer paint, what is very nice is that the imaging strands are not bright at all. So you can increase the concentration without having some drawback on your final image. As you can see here from the spectra, in red is the imaging strand. The signal is very low as compared to the blue curve, where you have the formation of the duplex, and you collect the signal from the Alexa dye. So we could demonstrate this in this imaging of microtubule HeLa cell experiment, and we could work with very high concentration of imaging strand, so which makes the acquisition very fast. So here, for example, 30 seconds only, and this you can also see from this FRC curve that your resolution is already very nice after 30 seconds. So we can compare with DNA paint and fret paint, and main conclusion is that we can get same resolution as DNA paint, but 60 folds faster. Compared to fret paint, we have similar results, but the design is much simpler. And okay, this is the first design, so there is still large room for improvement. So now I switched to the last topic. So here, in this one, we focus on using photon upconverting nanoparticle for single particle tracking microscopy. So such kind of system is very efficient in performing photon upconversion because the process behind this UCNP is energy transfer upconversion, which is a process that is many order of magnitude more efficient than two-photon absorption. And therefore, you can detect by your eyes the signal using only a continuous wave laser. So the person that visited our lab could have some opportunity to see it by their own eyes. So in the context of microscopy, why it is interesting is that because due to the fact that you need only low intensity to observe UCNP, in fact, this photon upconversion is a very specific features of UCNP, so you can detect them with nearly in zero photon background. And another interesting feature is their very high photostability. You can image them over hours, even days. Small size and sharp emission bands are also very interesting. So as compared to the typical label used for single particle tracking, So we think UCNP is a very good alternative, for example, to QDs that are blinking or dyes that are bleaching. So we have a nice collaboration with Thomas Hirsch that provides a very good monodisperse UCNP, an expert in the domain. And we made a big work with him in order to go from the raw UCNPs that are hydrophobic to solubilize them in water and provide them some targeting element. So here you can see on this star sky, it's immobilized UCNP on the cover slide, and we are very happy by the high homogeneity that you obtain. This is a condition to use them for application on cells. So on the cell, we selected a model cell system, which is the tracking of the high affinity receptor for the IgE. So by mixing our label UCNP, putting our label UCNP to the cell, we are able to, as you can see on the video, track, it's nearly live video, the motion of the receptor. And adding some antigen, we could probe that the system is still active because we have clustering of the receptor, slowdown of the motion, and which lead at the end to the degranulation of the cell. So we did comparison between what we obtained with UCNP, dyes, quantum dots, and here I guess you can clearly see that the signal for dyes is very little and it's photo bleach over time, and QD are very famous for their blinking, which limit in this case the length of the trajectory. So I will go very fast to this. So we make all the analysis, and finally we get, so one of the drawbacks of UCNP is that they are not very bright. So localization uncertainty is a little bit worse compared to the dye and QD, but what you have to keep in mind is that photon upconversion is specific to the UCNP. So here we make the experiment in HBSS that is completely transparent, but if we would have performed the experiment in some more complex medium like phenol red medium or I don't know, maybe blood, we would have obtained exactly the same signal with UCNP, rather than the signal with Alexa 647 of the QD will be very bad and will not obtain the same result. And anyway, the longest trajectory we obtain with UCNP because they're perfectly photostable over time. So I would like to thank bioimaging and the people involved in the bioimaging and to organize all this. So it was a pleasure for me to meet a lot of people. So Edouard Bertrand, and still not meet Caroline Thierry, with whom I had a lot of exchange. And of course, I would like to thank people from the Laboratory of Bioimaging and Pathology and the member of the PeakQuest platform. And the local organizer, of course, and you for your attention.
Thank you for introduction, and thank you for allowing me to present the research we're doing at the R&D team in the Normandy node. So it's how do I switch? Okay, sorry. So we are located at the University of Rouen, and we are a team of 6 permanent staff, mostly involved in the development of fluorophores for applications in bioimaging. So we are mostly synthetic organic chemists and photophysicists. So I will give you some example of what we have been doing in the last years. And we can take our inspiration from nature. And we were interested in this. Oh, the resolution is bad. Sorry. Oh, hope it will not be like that. So we were interested in this natural compound, which is called epicocconone, which was isolated from this fungus, Epicoccus nigrum. So epicocconone is not fluorescent by itself, but after binding to proteins, it becomes highly fluorescent. Oh, Sorry, I don't know why. So we made several analogs of this natural compound, and this one with this naphthyl side chain proved to be much more photostable than the natural compound. And by changing again this side chain to a fluorine moiety, we could improve the two-photon absorption cross-section up to 270 Gauss-Meyer. So this compound is useful for protein cell staining, and we could, you could, of course, excite it at 1 photon or 2 photon, depending on what you want to do. And you can see plasma and nuclear membranes staining, vesicle-like structures and filamentous structures in the cytoplasm. We made also near-infrared analogs of the parent compound by introducing this hemicyanin side chain, and we could have compound emitting at 730 nanometers. So this compound 11 is here in cell staining is the red dots here, that they do not colocalize with CM dial, neither with UXT. And you can see on this slide that they accumulate mostly at this portion of the cell. So we are also inspired by reaction discovery, and Cyril Sabot discovered a fluorogenic reaction where this benzoyl quinoxalinone is not fluorescent, but after irradiation you have cleavage of this bond, and then you can capture nearby biomolecule. So he developed this photoaffinity probe for membrane staining. And after incubation, so this dye is not fluorescent before irradiation, but after irradiation, you can see the membranes of the cell that are stained. And you can see also intracellular vesicles that are stained. Thibaut Galaverdin is interested in the indazole-based fluorophores, and he developed fluorogenic, these fluorogenic probes with this cation here. So of course, because it's cationic, it goes into the mitochondria. But once in the mitochondria, they aggregate, and there is a huge increase of fluorescence in the mitochondria. You can see also it's not fluorescent in water, but when you increase the concentration of sodium chloride in the water, they precipitate, they aggregate, and they become fluorescent. So you see the colocalization with MitoTracker. Also, if you change the substituent here, you can use this BTD heterocycle, and in this case, this compound goes to the lipidic compartments, and they also generate singlet oxygen. In addition to making our own fluorophores, we can use commercially available fluorophores with fluorescein in this case that Cyril Sabot linked to comptotecin, and they injected this probe in tumor, in mice tumor. And after histological examination, you can see that the probe accumulates at the border of the tumor and near the injection site. More recently, Sébastien Ballieu developed probes for glycerophospholipids, and they compare. So they made these probes starting from tartaric acid. So you have an extra hydroxyl here that can be functionalized as an azide for click chemistry. And so they have a synthetic probe that they compare with the commercially available probe, where the fluorophore here is at the position where fatty acid is located usually. In the case of the synthetic probe, you have the two fatty acid side chain, and the fluorophore is outside of the fatty acid side chain. So they made giant unilamellar vesicles, GUV, by mixing phosphatidylcholine and 4% of the probe, which is in green. So in this case, you see the GUV with the commercially available probe, or here with the synthetic probe. So by addition of chromogranin A, you can see that the synthetic probe clearly interacts with chromogranin A, and we can see the membrane remodeling and vesicle budding. You don't see such effects with the commercially available probe. Also, if you can also follow internalization of this glycerophospholipid probe, here in this case stained with ATO, and you can follow as a function of time the internalization. And so in the whole cell, the fluorescence in the whole cell is in blue here, and in green is the membrane, and you see that there's perfect, almost all the fluorescence is located in the membrane. Sorry for the quality of the image, I don't know why. So the current projects for bioimaging, we are currently developing solvatochromic dyes that have a super solvatochromism, sometimes more than 200 nanometers. We develop body Ps and other body Ps, photo switch, and theranostics probes. And we like to, in the future developments, make dyes for film instead. Of course, we are open to collaboration. And we acknowledge deeply the Primacen imaging structure led by Ludovic Gallas and Magalie Bénard and Damien Chapman. And I would like to thank you for your attention.
Thank you, Marc. Please.
Sorry, I'm not a chemist. And okay, thank you for the opportunity to present the roboscope. So I present that as a smart and fast microscopy for generic event-driven acquisition. So, how it works? Ah, no, this one. Okay. So today, when you do microscopy, you need to prepare fluorescent cells, long acquisition time. You need to stand in front of your microscope, supervised microscopy methods. So you need a lot of post-acquisition to sort your cells or your image, to qualify it. And then at the end, you address the scientific question. So the main idea is to do that during the acquisition. So once you get your fluorescent cells, you are able to detect and acquire autonomously qualified images. And then after that, you can go directly to the scientific question. So this is what everybody wants to do. Okay, so we have developed a system to control the microscope in the lab, and then now it's as you may know, it's a company in Scopper that make the control of the microscope. And then in this process, there is a particular aspect of the fact that you have an embedded system that makes all the control of all the peripherals of your microscope to go as fast as possible and to optimize the control of the microscope. So that means that your computer here is only a user interface computer, and there is no any direction during the acquisition process, which is directly driven by this kind of microcontroller. So the main idea of the roboscope is to do the same thing. So that means that we want, during the process of the acquisition of the image, we want to add another embedded system that is able to analyze in real time the image, so real-time image processing, and then to be able to feed back to the driving unit directly here as another embedded system. So the main idea is to go into this embedded system, and this is the roboscope project. So that means that I will go more in details into the process of the real-time image, because we want to segment individual cells after acquisition of, let's say, a huge field of cells, and then we want to classify individual cells. And as we classify individual cells, if we find the good cells, then we do a decision about individual cells. Okay, in terms of segmentation, I will go fast because the main idea is to make vignettes of cells after this wide field images. So you can do using conventional threshold segmentation, but you can do also using other kind of algorithm. In terms of classification, it's very interesting because we have some constraint for that. We want to be able to do some learning of image with a low quantity of labeled images. And we also want to go fast in terms of execution of this classification during the process of the real time. So that means that we go much more into some kind of neural network that are very little neural network able to do this kind of things. And in terms of training with a low quantity of image, what we do is we go into this semi-supervised generative adversarial network. So I'm not a big specialist of that, but the main idea of that is to be able to do, in the same time, the training of cells that are labeled images, but also on unlabeled images that are easy to get because you just acquire the image, you don't have to label that. And there is also a generator of fake image. And this generator wants to fake this discriminator. So this discriminator is, in fact, trained to don't fake this. And you use exactly the same discriminator for this fake real and for this the labeled images. And you do in the same time the training with these two with the same things. Once you do the acquisition, what we do is that we just use this classifier, and we use this classifier to classify the vignette that we get just before. Okay. So we did that. We do a classification with some MitoCheck database that is directly on the net. The main idea is to be able to recognize mitotic cells, in fact. So interphase, prometaphase, metaphase, anaphase, and apoptosis. And with few of that, so we trained that with 400 labeled image and with 148 unlabeled image, which is not a lot of that. And we were able to get an accuracy of 86%. And then after that, the main idea is to go with image that we acquire by ourselves because these images are directly in the net. So we do a full retraining. So we start with exactly the same network as before, but we do a full retraining with, for example, Zeiss database that we acquire with something like full retraining with 945 labeled vignettes. And with this, we get more or less the same accuracy. So we are able to go from one domain to another domain. And in fact, what we do also is that we can adapt the model using fine-tuning or transfer learning. The main idea is just to, let's say, fine-tune or transfer learning the last layer of the network. So that means that this network does not change, and we change only this one. And in fact, we are able also to change the quantity of the labeled image. Okay, so we use that with a Leica database, which are the microscope that we use for the roboscope, and we were able also to get an accuracy of around 80%. You say, okay, 80% is not so good. That's true. It's not so good if you want to, let's say, to be sure that you have an LT things. But if you just want to do some driven event, so probably 80% is sufficient. So it's not so bad to. So you lose some event, that's true, but you are able to get the events of interest. Okay, so now we have the algorithm. This algorithm is a little network. This little network can go into the embedded system, and then we can do in real time the analysis of the image that we acquire, and we can do a decision about individual cells. And this main idea is also important because if you think what we do is that we do automation of acquisition of image, and you can do that using the Nikon Jobs system. You know, it's possible. But if you think about all this system, it's a sequential system. So that means that you have acquisition, then you have analyze, then you have acquisition, then you have analyze. Once you get the analyze, you have a second acquisition, and then you do that sequentially. So the main idea of the system of the roboscope is to do that in parallel. So that means that you acquire your image for the modality 1, and then during the acquisition of the modality 1, you analyze in real time in parallel on another thread, you know, and this is possible. So then you do parallelization, and then so that means that you are, once you get one event of interest, you do the interruption of your sequence. So that means that you need to install an interruption of sequence. You have the first sequence, once you get the event of interest, up, you interrupt and you go to the second sequence. And this is the main idea, to go as fast as possible, and for example, to catch rare events. Okay, so how it works in terms of the application that I will present you. We have a UXT acquisition with a 20x at different positions. So we do a big screen of all our cells. Once we get all the cells, during this acquisition 1, we do the analysis with our algorithm that recognize, let's say, metaphase. And once we get a metaphase event, we interrupt the sequence 1 and we go to the sequence 2. And the sequence 2 is the change of color. Then we change also the objective. We center the cells of interest. And then we do a longer acquisition during 6 minutes. It's not a long scan of acquisition, but it's a specific acquisition. And this specific acquisition is to do and to characterize the dynamics of the spindle, mitotic spindle. So the application is to do that with non-treated cells, asynchronous cells, in which the metaphase is a rare event. Okay, so you need to find the metaphase. And we do that with synchronized cells that we do with a double thymidine block. And this synchronized cells, there is more mitotic event because we are at the good mitosis. But also, it's probably a transform event, and we want to see if there is a change into the dynamics of the mitotic spindle that we acquire during the process of the roboscope. And in fact, these are the images. What we follow is that we follow the motion of this pole, and this is more or less the work of my colleague and collaborator Jacques Pécréau. He was able, by measuring the length and by measuring the variation of the length during the process of the metaphase, he was able to do a Fourier transform analysis. And with this Fourier transform analysis, he was able to know how he's doing the mechanics of the spindle during the process of the mitosis. And then he was able to see a difference in double thymidine block and in non-synchronized cells, as you can see here. So that means that when you synchronize your cells, so you make a fake into the division of the cell. Okay, in conclusion, semi-supervised GAN-based method to be able to have a small training set. And the SGAN can easily adapt to change a different cell line or marker. We have also some other things in which we can do a change in microscopy modalities. And it's interesting because we think about to have a kind of generic approach for that. And the roboscope enables investigating spindle mechanics without synchronizing the cell, which is nice. So these are the people working on that. So this project is in collaboration with InScoper that go to embed the train model and the automation. Also in collaboration with the team of Jacques Pécréau, in which is involved into the design of a machine learning algorithm application to cell division. And with my team, we are in the data preparation of training all these applications to biology, the support, and we are recruiting at that moment because we have money at that moment. It's nice. And then we our work, we think to have an applied mathematician for the development of artificial intelligence algorithm, and microscopies for biology application, or somebody which can do both. Thank you. Thank you.
So we have a very colorful presentation. Thanks.
Hello, everyone. Today I would like to present split SMLM, which is a spectral demixing method for high precision multicolor localization microscopy. Yes, so this method is available at the Integrated Structural Biology platform. Oh. Okay. Yes, at the IGBMC. Now, this work was mainly done by Leonid Andronov. He was responsible for super resolution microscopy before my arrival at the IGBMC. And so this is a short introduction into single molecule localization microscopy, or SMLM. I think you're all familiar with it. So compared to a typical wide field fluorescence image, the idea here is that most of the fluorophores will reside in a dark state, and only a small subset of fluorophores will be in an emissive on state. And in this case, if we have non-overlapping emissive point spread functions, we can accurately localize them, and then by sampling many such sparse subsets over time, we can then reconstruct a super resolution image. And now there are many ways to switch fluorophores on and off, which is also called blinking. So we will, in our case, we will focus on storm imaging, or which is based on the switching of fluorescent dyes, or particularly we are using direct storm, which where we are using special imaging buffers to modulate the blinking behavior of fluorescent dyes. Now, if one wants to perform multicolor SMLM, there are two possible approaches. So the common approach would be based on sequential imaging of spectrally different fluorophores. So one would require a full imaging stack for fluorophore 1 with laser 1, and then fluorophore 2 with laser 2, and so on. But there's also the ratiometric approach, which is based on simultaneous imaging of spectrally close fluorophores. And so this approach has been around for a while, but I will focus here on our implementation, which we call split SMLM. And now key to this approach is a dichroic mirror in the imaging path of the microscope. So we split actually the emission into two channels on the camera, a long wavelength channel and a short wavelength channel. And because the fluorophores are spectrally close to each other, they will show up in both channels, but with a characteristic ratio. And from this characteristic ratio, we can then assign the fluorophore type, which is a process called spectral demixing. So you can see here in this bivariate histogram that we can separate different fluorophores, and you can also see that we carefully tuned our setup for a specific fluorophore triplet, which is CF680, CF660, and Alexa Fluor 647. Now, what are the advantages of ratiometric imaging? So first, it's imaging speed, because one has to image only with one color. The drift is same in both channels, so this simplifies the drift correction, and we have spectrally close fluorophores that provide equivalent data quality. This is an important point because in storm imaging, fluorescent dyes that emit in the far red are considered the best from their blinking characteristics. So we can use only red fluorophores. Then we can also we can remove non-reliable localizations. For instance, if there are localizations in one channel due to noise that do not pair in the second channel, they can easily be removed. So there's no detections due to noise. Chromatic errors are of course reduced, and the excitation path of the microscope is simplified. So we need only one laser in principle and less optical components to image with one color. Channel alignment is also simplified. Here we can use simple alignment procedures such as cross-correlation to align the channels, so one does not have to rely on fiducial markers for channel alignment. So we did several refinements compared to previous approaches. So experimentally, we optimized for our setup for 3-color imaging with an optimally adapted set of filters and dichroics. We also optimized the imaging buffer to obtain a similar blinking behavior of all the 3 dyes. On the software side, we maximize the localization precision by using all the detected photons from both channels. We also correct for residual chromatic distortions, and we also refine the coordinates from relocalization events that can occur in SMLM. Now, all these refinements in the sum, they will improve the resolution. So we obtain, this is an image, those are images for nucleoporine 96. And so we can obtain a resolution of 20 nanometers here, but it needs to be kept in mind that these images were obtained with secondary antibody labeling. So there is some inherent lowering in the resolution only due to the degree of freedom of labeling. So we have developed a dedicated software to perform SMLM data analysis. So here the general framework is SharpVisu, and it can perform general SMLM data processing such as drift correction, refinement of localization events, or measuring the resolution. And then there is also a plugin which is called SplitVisu, and it can be used for spectral demixing. It allows to select input, to select the input regions, and then it performs channel alignment, fluorophore pairing, and then fluorophore demixing, and also corrections of chromatic errors. So this shows an application of the method for 3-color imaging of the nuclear pore complex. What you can see here are averaged MPCs. So for instance, we find that nucleoporine 62 localizes to the center of the channel, and we can see this typical 8-fold rotational symmetry for nucleoporine 96. On the other hand, lamin B1 is absent from the nuclear pore. And for instance, for another nuclear pore protein, POM121, we find that it forms irregular ring structure at the nuclear pore envelope protrusions inside the MPC, but without clogging the channel. Another plugin that comes with the SharpVisu software is ClusterVisu. So it can be used for Voronoi tessellation to visualize and quantify protein clusters. So usually in cluster analysis, a threshold is required, but in this case, the threshold can be obtained from Monte Carlo simulations, which allows for automated segmentation. And we can also, from the Voronoi cells, we can obtain density maps. And these density maps then allow for density-based segmentation. And this concept can be extended to 3 dimensions. And here we show 3-dimensional segmentation of nucleosome clusters as an example. Now, to summarize, we have refined or improved the SML data quality for multicolor imaging. We have developed a dedicated software, Sharpisu, which integrates all the necessary processing steps into a single interactive tool. And the applications illustrate that we can perform high-precision multicolor localization microscopy. And also importantly, it only requires commercially available components, so it's easy to implement also in biological laboratories. And this method is available at the Integrated Structural Biology platform at the Center of, hosted at the Center of, for Integrative Biology at the IGBMC. Okay, short outlook. So I'm currently working, besides other projects, on extending this concept to 3 dimensions based on astigmatism, and also to include a deep learning framework for 3D emitter localization. So with this, I would like to thank all the people involved, and I'm glad to answer any questions.
Yeah, thanks for the introduction. As it was introduced yesterday, and you have seen it from the various fields, fluorescence is the queen of the battlefield in microscopy, so we do something else. Basically, it's unlabeled imaging. We are a small team. Matthieu Sainlos is our engineer making the true developments. He's over there. Nicolas could not come, and we have PhD students. I will present their work afterwards. So basically, when you don't want to use labels, there are not so many solutions. In vegetal science, for example, autofluorescence works very well. Unfortunately, it's not so good when dealing with animal cells. Another possible technique is harmonic generation, mostly second harmonic and third harmonic generation in tissues and cells. These are images from the LOB in Palaiseau. The interesting feature of this technique, it's a kind of morphological approach, and the information you get from second harmonic generation or third harmonic generation is complementary and very different, because the involved physical phenomena are very different. And the third possible approach is so-called CAS microscopy, coherent anti-Stokes Raman scattering, which also provides a different information from, for example, second harmonic generation. These are images from colleagues in Marseille. CAS microscopy is also interesting because it's highly linked to the chemical compounds. The drawback of these techniques, they involve the femtosecond lasers, very high-speed instrumentation, etc. They are quite bulky, quite costly, and the interpretation of the images is not so easy. So if you really want to simplify the stuff, there's no way. You come back to the traditional transmission microscope. And in fact, if you look in history, there have been dozens of techniques developed to improve both the image quality, the sensitivity, the detection, to add new modalities like polarization microscopy, which gives you a new contrast. These are the, I would say, the historical development, but it continues, for example, near here in Freiburg, the so-called ROX microscopy has been developed, which is a dark field, a very fast, I would say, optimal resolution approach, which has been recently developed. And if you analyze how this system works, in fact, they are characterized by some control of the illumination. So, for example, in the ultramicroscope, you illuminate from the side. Some understanding of what happens when light interacts with the phenomena. In polarization microscopy, it's rather easy compared to the other techniques, but you get also you can get quantitative information from a DIC microscopy, for example. Drawback is they are 2D, and all the specimens we want to image are in reality 3D. So 3D imaging, when you don't make optical slices, this goes back to holography. So holography is an old technique. In transmission, it works like this. Because we can detect only intensity in the optical range, you have to mix the wave, which is diffracted, deformed by the sample. When the plane wave goes through, you have to mix it with a reference wave. So it's a modulation. And of course, the problem is to demodulate. This is what you record. And in fact, you have an intensity image, which combines the illumination and the reference. But you have two so-called orders of diffraction which are mixing the information you are looking for and a reference which you know. And that is in radio wave, it's simply demodulation, frequency modulation, etc. So in optics, it was a little bit more difficult, and Denis Gabor proposed the technique. And it happens that a few years after he proposed the technique, there was this seminal paper from Emil Wolf explaining how to reconstruct the object from holographic data. But at that time, no computer, lasers were just emerging, no CCD, so the technique was lost. But, of course, discovered again and adapted to digital holographic microscopy. That was in the 90s, mostly at EPFL. They were the pioneering. So you just squeeze the previous setup within a microscope. So you illuminate with your wave. You detect both the illumination wave and the diffracted wave in red here. So here the colors are not related to fluorescence wavelength. It's always the same wavelength. You mixed everything with a reference wave. So you have a 3-wave interferometer. And you record and you reconstruct. So I will not enter into the details of reconstructing, but you can get very nice images. However, when you look at the 3D image you get, this is what you get from your sample. In XY, it's not so bad, but along the Z-axis, it's terrible. And the reason is easy to understand. If you remember, we are violating an old principle of microscopy, which is you have to use a condenser and illuminate the sample from all the angles. This is not possible with coherent wave because it would create a confocal spot, and that's not what we want. So it works XY, but not so well in Z direction. That's because it's a transmission setup. In reflection, it's okay, but in a transmission setup, it's not good. And in fact, it's not so quantitative also because basically you integrate. You go through the specimen and you integrate this quantity, which mixes spatial dimensions because you integrate along the path when you go through, and the physical information we are looking for, which is in fact the index of refraction of the sample. So it works very well for many applications, but not so much for 3D imaging. So the trick from the Wolf paper was how can we come back to the condenser? Right? You are supposed to use a high NA condenser to have good ideas, good images in transmission. So that's the so-called tomographic diffraction microscopy, optical diffraction microscopy, synthetic aperture microscopy, phase microscopy, etc. It's known under many names. It's just an extension of digital holographic microscopy in which you now actively control the illumination over the sample. You can do this by rotating the sample, that's like in a scanner, but you can also tip tilt at the entrance of a high-NA objective so as to change the illumination. And here is an example of a movie where we demodulate the hologram, but you see that what is more or less a sphere becomes more and more elongated, and that's because it's when you incline the rays more and more. If we look at the series of images, we have bad images, and in fact, it's not so good a priori, but with properly image processing and information reassignments, a so-called inverse problem, we try to go back to the sample which generate this kind of movie. In fact, from a series of bad resolution holograms, you can have a nice 3D image. Here it's a pollen grain. It's not dead. So this is the equivalent of optical slicing, but it's purely numerical. And the resolution of such an image is basically twice the resolution of any of the intermediary hologram. And we have a through 3D sectioning. And it's quantitative because the gray levels you see here are directly linked to the index of refraction of the sample. In fact, the technique was invented way before. It's a technique which comes from astronomy, which is called aperture synthesis technique, and now it's known as aperture synthesis. It's easy in the radio wave range because you measure amplitude and phase very easily. It's not easy in the optical range because you have no direct measurements of the amplitude and phase, but thanks to holography, we can do it. So here is a higher magnification view of this pollen. First thing is we have a dual information because we measure both absorption and refraction. So these are two informations about the sample. Interesting thing, for example, here, it's obviously the color of the pollens, the absorption is restricted to the envelope. And on these images, analyzing the very small details which we could identify, we have a resolution of about 150 nanometers. Because we record the entirety of the physical information resulting from light sample interaction, in fact, we can recompute all kinds of transmission microscopes you may imagine. So multimodal imaging in that context means we can recompute a DIC, a Reinberg illumination, a dark field, or whatever you want. So here is, for example, an image of a simulated, a reconstructed dark field image which we would have obtained. It highlights the highly scattering parts of the pollen, for example, the spikes. The phase contrast is not so successful, but for example, the DIC is very good. We can make so-called optical coloration, so-called Reinberg illumination, because we can simply numerically assign the color to the various illuminations which we are using, so it's purely numerical. And we can even compute images which are impossible to make in a standard microscope, for example, a Reinberg DIC, because if you look at the optical train in a Reinberg configuration and in a DIC configuration, they are incompatible. So you would need two microscopes, but we can, for example, recompute this. There is a good news for the platform and for the people who want to use it. So there are now 3 companies selling such instruments. Nanolive in Switzerland, TomoCube in Korea, and PhiOptics in the US. They use a slightly different approach, but it's similar. And there are about a little bit less than a dozen of active groups in the world trying to further push this technique. So if it's commercially available, you would say there are many applications. I invite you to have a look at the websites of these companies. They make a good effort into collecting everything which is made and illustrating. And for example, I have seen yesterday something about lipid quantification. Lipids are very easily seen in tomography. Here is another example with nanodiamonds. It's even a higher contrast. But it has been applied to stem cell studies, etc., etc. So have a look there. And because we are a development team, we have to go further. So if you want to go further and try to bring back some advantages of other modalities, no choice to go back to the physics, for example, which brings you new concepts for instrumentation. And modern instruments, they don't deliver images. So of course, you are closely linked to computer sciences and reconstruction models, but also applications very often drive new requests, which implies modifying the instrumentation, etc. We recently made a review of the technique, so please have a look there. We all need citations. So thank you. So now we have two projects currently. One of the problems is all the microscopes have a non-isotropic resolution. So we want to further push the technique to isotropic resolutions by developing 4-pi microscopy. I will not enter in the details, but basically it's combining transmission and reflection. And Carlos has a poster over there, so please go there. Another demand is to bring back chemical sensitivity, and chemical sensitivity, you can have it by spectroscopy. So thanks to the Separary Maprogen, we will make an hyperspectral TDM. That's the PhD topic of Leonardo, who also has a poster there. They have to work. And this is a long-term project which we have with IS2M, also in the node, to study inflammation induced by plastics, silicone particles, when they are captured by macrophage. With IGBMC, here I entered the minefield for me, so I will not go there. But they want a combined confocal fluorescence and tomographic setup. So we have developed such a system which has been installed at IGBMC. So here is the fluorescence part. Here is the holographic part, tomographic part. It works. Unfortunately, it's not very final user-friendly. So thanks. That's the good news of the story and of the node is that we got a grant from France-BioImaging to modify, and these images are from last week. So they bought a regular, I think, Leica system, which has been modified, the illumination column. They modified it, and now we started reinstalling the setup, and that is made with, for the biology, it's a Marc Ruff's group with Julien Baptiste and Didier Hensch and Victor Piolo-Demes for the modification and essentially the mechanical transformation of the system. So the story goes on. Thank you.
Thank you, Aliénor.
Hello, thanks for the opportunity to talk today. I will introduce a method to measure light intensity, which was part of my PhD work in Ludovic Julien's lab at INS in collaboration with the Sony Computer Science Laboratories. I will begin by the context. Why did we formalize this method that was already present in the literature somehow, but we tried to make it easy to use? It's because we were working on single-cell microalgae. Oh, disclaimer, for the talk, the images have problems. It's to highlight the issues I'm talking about, so it's on purpose. So we were working on single-cell microalgae, and as Olivier said, they emit autofluorescence, and this fluorescence carries physiological information related to photosynthesis. So when we have an image like this, for example, we see two colonies, And we want to know the origin of this difference of intensity. And if it's physiology, for us, it's interesting, but it can also come from a different heterogeneous illumination of your sample or a problem in your optical transfer function. And when we study single cell, we study dispersion, so we really need to identify sources of heterogeneity. And so just to be precise, when I talk about light intensity, I mean power divided by area. So it's irradiance, illuminance, and if you talk about photosynthesis research, it's number of photons per area per second. And so currently, if you want to calibrate a light intensity, what you do is you measure a power, for example, with a power meter, and you divide by the area that you can measure, for example, with a microscope. So it gives you an average light intensity, but here, for example, you see it's quite heterogeneous. So a suggestion proposed is to use an homogeneous fluorescence slide, and then you say, okay, my heterogeneity reflects the heterogeneity of illumination. But is it really the case? Because what you do is you illuminate at the sample, and you image with the camera. And so the heterogeneity you see here can come from what's happening at the sample illumination, or from a transmission function of your imaging system. So you cannot discriminate the two. So what we introduce is just, we use fluorescent actinometers, and the principle is a molecule, these are molecules that undergo photoconversion when exposed to light. So I'll take the example of here of fluorophore that loses fluorescence when exposed to light. And so you turn on the light and gradually lose the fluorescence. And this is this gradually part that we'll be interested in, really looking at the kinetics of the evolution of fluorescence with time. And so for the rest of the talk, I will focus, for example, on this photo-switchable reversible fluorophore, which loses fluorescence when exposed to blue light. And you see the kinetic constant of this conversion is proportional to the light intensity. And so if you do your experiment, you can measure the decay of the fluorescence, and you're able to fit the curve, retrieve this kinetic tau value that you can relate to the light intensity. So just in practice, for example, this one, you excite it in blue and it emits in the green. It's soluble in water. You can have it by genetically encoding and extraction. And it works in a range of 5 orders of magnitude. And in practice, for the lowest intensity, the experimental duration is 2 minutes for your light calibration. And we have another proposed set of other molecules depending on other wavelengths. And it's also interesting because it's actually a reversible fluorophore, which means if you shine purple light, it will retrieve the fluorescent state, or if you wait for a few minutes, it will come back to thermally to the fluorescent state, so you can reuse it several times when you're trying to do several calibrations. So in practice, say you have an instrument you want to calibrate, this would be your experiment. You have your light source, your detector. At the position sample, you put this actinometer, measure the fluorescence decay like this, and then retrieve the light intensity. And you do it for different settings of your microscope, and you're able to associate your percentage on the microscope setting to the light intensity. You decide for the units that you're interested in. So if you want to apply it in practice, we have disposed in a GitHub repo the code to process your data if you provide as a CSV file the evolution with time of your response. Now we can also do it with a microscope and to see the uniformity of illumination on the microscope. And the experiment is pretty much the same. You just have to make sure that you have not a small concentration of the fluorophore to make sure that the intensity is homogeneous in the sample. And another step is not so easy to focus because, as you see, we lose the fluorescence quite fast. So, for example, you can add purple light to shine back during your focus. But in practice, you would get a video like this. I will talk later about why the diameter is still shining, but it's related to diffusion. So you take your video, and you have, for each pixel, you retrieve the light curve, the decay of intensity. You have a map of tau values that you directly convert into light intensity with the formula. And here, it's an image that we took by patterning the light illumination with an old argentic photography. And this is what we get on the camera. And for example, along the gradient, you see that there is, along the red line, there is a gradient. And here, it's what we get when we do the processing. So first, we have the light intensity at every pixel, but also we identify that along the line, there is no gradient. So actually, the heterogeneity we would see on your sample did not come from the heterogeneity of illumination, but really from the optical setups. So it's possible to make the distinction. I don't have, maybe I have time. I spoke fast. We also showed with a confocal microscope that you can do illumination in depth. So what we did is we took, maybe I have the slide. No, I'll see. We took a pad of agarose and put some fluorophore on top, did the measurement, flipped it, and did the measurement again. So at between the two experiments, the difference is that the light had cross absorbing a sample. And we see that a 200 micrometer thick sample. And we see a difference in the light intensity for this semi-transparent medium. And we also propose a method so since the one I presented here works with blue light, but you may want to calibrate another light, but don't have access to DROMPA2, which you have to express with bacteria. We propose in the paper a protocol to transfer the information from one wavelength to another really easily using an inert fluorophore that absorbs in the whole range. And so to come back to the defaults of the method, one of the limits is the molecular diffusion. And so this is what you see on the camera if you prepare your liquid sample versus liquid sample embedded in a gel. And then we do the video analysis and processing. And this is the intensity map you get with the liquid versus with the gel. And so you see with the liquid, while the experiment is running, the molecules are receiving different light intensities and you get blurred information. But when you embed it in a gel, you see that the distribution in values are similar, but you remove this effect of molecular diffusion. So to summarize, we introduced the method to do superficial light intensity measurement. We're able to have access to the distribution of illumination really at the sample, and with a high signal of a noise ratio and different wavelengths and depth distribution. Sorry, I mixed up the timing.
Hello everyone, I am Mrinal, a postdoctoral researcher, and he is Yuri, a PhD student. So we both work with Ludovic Julian from ENS, Paris. So today we'll talk about non-covalent reversibly photoswitchable fluorescent protein. So recently we have developed protein tags which generally remains non-fluorescent in solution state. However, once it binds with the protein, it becomes fluorescent. And we could see the large shift, red shift of absorption maxima upon binding with the protein. So recently, now we are interested in developing reversibly photoswitchable protein tags. So the idea here is to is to illuminate the bound state with the protein tag, and idea is to photoisomerize upon illumination. And once it is the protein tag gets photoisomerized, it will have change of binding affinity with the protein. So for that reason, it will go out of the cavity of the protein. So like that, we can switch between on and off state, which this process could be very beneficial for dynamic contrast imaging, as well as in super resolution imaging also. Okay, so for this we need to develop some fluorescent or the protein tag which can photoisomerize very fast, and it should change its binding affinity upon photoisomerization. Another application of this process could be photoacoustic imaging, where we will read out the structural change of this protein tag, and this could help us in depth imaging and overcoming the light scattering. Now, so far we have this fluorescent could be synthesized very easily in a single step, and so far we have developed a series of fluorescent emitting across the visible region. Okay, so yeah.
So we have a library of fluorescent, and the next step is that we screen them for photo switching or photoisomerization properties in fluorescence using our homemade fluorimeter setup. And so we identified a number of fluorescents that had good photo switching properties. For example, this one, HBr3Cl, which upon illumination with violet or blue light would go from the Z isomer to the E isomer, and at photostationary state, we have about 50% of E isomer. One important aspect is that this process is reversible thermally, so in darkness. And in fact, it's quite fast. It takes around 10 to 1 minute, 10 seconds to 1 minute. So, fluorescence is great for measuring kinetics, but it doesn't give us access to the concentration or information about the structure of the species. So for this, we wanted to go to nuclear magnetic resonance spectrometry. And here we start running into problems, because for NMR, we need to be in high concentration. And since we have some samples that quickly equilibrate thermally, it means that we are forced to do in situ illumination during the experiment. And moreover, the illumination needs to be homogeneous in the whole volume, otherwise we will not see anything useful. So for this, we had to develop a device for 3D homogeneous illumination, which is compatible with NMR. And not to go too much into detail, but basically it exploits diffusion of light through a thin sample to maintain uniformity. So to calibrate and characterize it, we used the DROMPA 2 actinometer that my colleague talked about, and to obtain 2D maps of illumination and calibration curves. And we coupled this with some extensive optical simulations using ZMAX to prove that we have indeed a robust system for 3D homogeneous illumination. And so using this device, we studied the three fluorescents first, and we see that as we expect, when we increase the intensity of light, we see that we start from only the Z isomer, but the more light we have, the more we form the E isomer. And so this is quantitative. So next up, we collaborated with Bernard Brucher at the Institut de Biologie Structurale at Grenoble to see the interaction with the protein. And here we see that we indeed, when we apply light, we form a new complex, the E complex. And if we look at the most affected residues, it's the ones that are really around the fluorescent. So it's really a local effect. So in order to interpret all of this data, we had to develop a four-state mathematical model, which has quite a few degrees of freedom. But basically, fitting all of the data into this model, we can extract the most important parameters, which is the binding affinities of the two isomers, which are not easy to measure directly. And we see that, in fact, we go from 0.1 nanomolar to 200 nanomolar when we photo switch, which is like 3 orders of magnitude affinity difference. So in fact, the protein is quite selective for the correct isomer. And so finally, to conclude, we managed to find fluorogens that have good change of affinity upon photo switching. This required us to use different techniques and some mathematical modeling. And so now we are kind of in the exciting phase of research where we are applying this in confocal and epifluorescence microscopy in cells to explore this phenomena of change of affinity to do some dynamic imaging. So, and as my colleague said, we also want to apply this for super resolution, photoacoustic imaging, and also for optogenetics. Thank you for your attention.
Okay, so thank you. Good morning to you. So, as you said, I'm from the Bordeaux node. So I work more specifically at the INES Institute, Interdisciplinary Institute for Neuroscience. I work in the team of Daniel Choquet, and I use a lot the proximity of the Bordeaux Imaging Center facility, which is a big part of FBI. So what am I interested in? So essentially, from a global point of view, I'm really focusing on trying to develop tools to label proteins or to modulate their activity, and I'm really, really interested on working on endogenous ones, so trying to develop system for that. And I do that in the context of studying neuronal cells and more precisely synapses and their proteins. And so it's just to illustrate a number of the challenges that I am and everybody that's doing that facing when you want to label protein in such complex systems. So you have high densities of cells in tissues when you want to study really integrated models, or even when you go to smaller details and synaptic compartments, you really have crowded environment with plenty of protein, and you have to be able to reach and label efficiently those proteins, especially when they're endogenous. So we're going to focus on all the effort that we're making to try to develop and improve our capacity to label proteins. And we have essentially two strategies. One is to focus on endogenous ones by the development of binders that we, in the end, use as intrabodies. And then the other one is trying to develop more generic systems that will work either using a tag and a small probe, in particular for CLEM application, and also, but I won't mention that today, developing new fluorescent, phototransformable fluorescent proteins. And for that, we essentially use protein engineering strategy. So I'll present that a bit later. We use phase display, we use any other strategy that we can use or methodology that we can use that can be found in protein engineering field. So I'll present you some examples of what we do, what we can do on endogenous protein. So in this case, we use directed protein evolution. We try to target neuronal proteins of interest, and we use that for modulation, or in this case for imaging, and we've done the two. So we focused on one of the main synaptic scaffold protein, PSD95, which has one of the particularities that it belongs to a larger family of paralogs, so with very similar or high sequence conservation. We've applied phage display strategy to isolate from library that we make binders of some of the domains, and we've been implementing strategy to really be able to isolate binders that are really specific for one of the, so PSD95 in this case, so one of the protein in the paralog family, and we also managed to develop some for the other ones. We've teamed up with other teams, so with the BIC, with structural biologists in Bordeaux, to try to really characterize those tools. I will spare you that, but it's a lot of work. But eventually from that, especially when we understand how they bind, their binding kinetics and their specificity, we can really push forward the tools and try to use them to actually label endogenous proteins as intrabody. So we fuse them to an element that's going to be fluorescent, express them in cells, and then label the protein that was not genetically modified in this case. So to make it really a short story, we have a system that allows us to regulate the level of the intrabody to the level of the target by fusing repressors, zinc finger, and fluorescent proteins. We've made plenty of characterization and made sure that our system allows us to be really correlated with the level of expression of the protein. It matches what we can get with antibody and outperforms them eventually. And it's totally specific because when we knock down the protein, we have no more signal, as you can see in this case, so, except for the nucleus because of the regulation system. But we can completely lose the system, so it only binds to our target. And with that in hand, we can do a bit more engineering, modify our probes, and then generate a system that can be adapted to advanced imaging techniques. So in this case, pile microscopy by using photoconvertible MEOS 3.2 proteins, and we can achieve high resolution imaging of those proteins and the endogenous one again. We can also apply that to STED, benefit from organic dyes by using, in this case, the SnapTag strategy. And we can also using the same strategy, we can also have access to DNA paints. So now we're working on other proteins from the same family, and we're trying to be able to use those in parallel so that we can really discriminate and localize precisely where all the proteins from the family are localized. Mixing up the buttons. So just to finish on that part, so we use directed protein evolution strategy. We can generate binders, apply them, and we've taken all the time, especially for the first one, to really validate the whole pipeline, to really make sure that we can go from the isolation of our definition of a target, selection, isolation of a binder, characterization, and then eventually use it as an intrabody to do either imaging or modulate. And now we're working on other targets. So that's one aspect. And so the other aspect, which I mentioned earlier, is try to generate strategies that are a bit more general and that are adapted for super resolution imaging. So I'll present two of those. The first one that we developed some years ago is relying on the use of a monomeric streptavidin, which then compared to streptavidin benefit from the fact that it's a monomeric system, much smaller than other systems to label small tags, and then the tag by itself is really only comprised of a short 15 amino acid that can be selectively biotinylated by an enzyme that you incorporate. Done that a few years ago, but the system has, even though that the system is clearly outperforming antibody to label proteins, as shown in our first studies, much more efficient, higher density of labeling, and better access to confined region. It still suffers some limitation, and we worked on those. So we tried to improve the affinity, the kinetic, the thermostability, the control labeling, and the brightness of all of the probe system. So with a gain of several order of magnitude in terms with the half-life of the complex that we form, much higher thermal stability. So we have much better and improved systems, and now we can push them to label endogenous protein that with a knock-in strategy, where we can directly label them in tissue, specifically in compatible with live imaging. We can go several tens of micrometers. In terms of imaging, we can do single particle tracking on all sorts of strategy, and that's really an improvement compared to the initial version of the tool. Another one, just to finish, another example is we're also trying to work now on the development of probe adapted for clamp strategies. And so we try to really, we first validated our system with a nanobody that targets the GFP. So we worked on how to make sure that we can have a single coupling of one small nanogold gold particle Currently, we're using 1.4 nanometer and a single fluorophore. And we validated that system here, for example, in a model system where we have a really tight contact that mimics synaptic contacts, where we have two proteins that are overexpressed and that make the physical contact between the two cells. And we can incorporate our CLAM probe, and that can get enriched at the region where we have the contacts, and we can see that we have the gold particle that can enter those regions. So now we're moving on to use those for labeling of neurons. And so just to finish, I've shown you that we can use directed protein evolution to generate novel probe or endogenous proteins. Now that we validated the whole pipeline, we're applying it to plenty of other proteins, collaborating with colleagues from around. And we're also trying in parallel to try to improve the tools that can be used that are more generic to do super resolution imaging or CLAM imaging. And so just to finish, so it's a teamwork with plenty of people. So I have to acknowledge the work of a number of people from the group and from collaborators that help us develop those tools. Thank you.
Thanks a lot for having me here. It's really an honor, and I realized that giving one of the plenary talks, I have quite a lot of time, in contrast to everyone who just basically briefly says, I have this tool, and I have this tool, and I have this tool. So I actually have the advantage of having time and to talk a bit more deeply about tools. And at the same time, I have also the problem that I have to keep you happy for quite a long time. So let's hope that works. I kind of brought 3 topics. So as you can see already from my first slide, so I'm a trained physicist, but for a long, long while worked on microbiology. So my first group leader position was at the Max Planck for microbiology already now, oh my God, 10 years ago. And even before in my postdoc and PhD, I mainly worked with microbes. So the examples you will see are from the microbiological world. And we try to do quantitative biology, try to understand the cell biology of microbes using a lot of things, using what everyone knows, like wet lab techniques, like a lot of tools that exist from biochemistry. But one of our prime tools, and that's why I guess I was invited here, is to use also single molecule imaging and single molecule tracking to then really see what happens on a molecular level within the living cells. And I'm not saying that these are the only tools you should use. That's why I want to emphasize we also back up a lot of data or do a lot of data with classic biochemistry and ensemble measurements. But there are some things which I think it's quite worth it to do the effort and to go all the way in optimizing to measuring single molecules in the cell itself while it's still living. And yeah, so the first example will be a structural study. The second example will be adding dynamics to it, so a tracking study. And the third one has to do with, and now I have to focus on the pointer, the reason why our group picture from 2024 looks that strange. So it's a new imaging sensor, the so-called event-based sensor. And I'm not sure how many of you have been at MIFI Bio. There was a workshop by Ignacio on this. So it's a cool new sensor. And so if you're bored with the 2 first parts of the story, maybe wake up again for the last version, because that's very, very general. It's a camera, right? Okay. So when using single molecule techniques for anything, like any biology you could possibly think of, you have to optimize it for your particular research question, right? And that could be very, very different things. And in our case, the things we struggle most with are cell walls. We don't have only cell membranes. We usually have cell walls. We have in total absolute low counts. So it could be that your protein of interest is there only in 50 copies in the cell. And this is a problem when you think about extrinsic labeling. The lowest level would be an antibody staining, right? And then the problem is A, you have to get it in, and B, you have to get everything which is not staining specifically out again, because you have then otherwise unspecific rest labels sitting around in the cell. And if this is more than 50, then you will see more background than you actually see your protein of interest. So this is a problem, and you really, really want to go to very low protein copy numbers. We have, compared to mammalian cells, much higher molecular crowding. So it's just more dense. We have a lot of different intracellular environments. So we have sometimes strange pH. We have a lot of charges around. So and that is also problematic in terms of different organisms. So the microbial world has, I think, the highest species diversity, if I ignore viruses for a moment. But if I just take microbes, then the diversity is big. You have all these extremophiles. You have all these pH ranges, the temperature ranges. Microbes even survive in vacuum, right? There are all these records of where microbes can actually thrive and survive. And that obviously, depending on what you're interested in, what you study, reflects back in a lot of different metabolisms and environments you actually have to deal with when studying them. Yeah, and that's also true for then, in the end, we are a fluorescence group, right? So for autofluorescent issues. And so basically, a lot of our work is also to establish single molecule imaging for different microbes. So not only the classic, so the HeLa cell of the microbial world is E. coli, right? So Escherichia coli is the standard model organisms most people use, but there is much, much more, and especially for strange environments, really interesting cell biology happening, right? So microbes that can survive interesting environments have interesting proteins, right? Interesting pathways. So there is a reason why you not only want to study everything in E. coli, but also go into more diverse scenarios. So this is the list of our microbes on the microscopic stage. So these are the ones we have tackled and have optimized protocols for. And I would say the one we are currently most proud of is this example. It's an archaeon. So it's not a eukaryote, it's not a bacteria, it's a prokaryote, but an archaeon, the third domain of life. And this particular archaeon here, it's one of the few ones you can actually kind of handle okay-ish in the lab. It's thermophilic, so it grows at elevated temperatures, which is already a problem for a microscope. It's halophilic, so 5 molar salt and plus. It has a high GC content for the ones that are doing cloning, that's annoying. And it has pigments. So it was quite a journey to get that on stage and get that working reliably. But yeah, and so for our very first proof of concept paper, and there's now more in the making, but the very first one, which is already published, we chose something where you all believe me if I say we did it specifically, which is the FDSZ division ring that always is in the middle of the cell and lines up in cell division. So I think that's why we chose it as a model protein to show it works, because here's not much debate about if we, imaged the right thing. And another thing which is tricky, and we saw already quite some talks that tackled this, is like correlative microscopy, combining different schemes, right? And we were, when it came up, also interested in combining fluorescence microscopy, especially super-res microscopy, with expansion microscopy, and a lot of groups did, and there are many, many versions to Rome on how to combine this. I want to show you the most funny problem we had. We had a yeast, so that's pombe, and this yeast cell, when we inflated it, so it is rod-shaped, yeah, so it should be rod-shaped, but when we inflated it for at least half a year, it remained this really nice thin tail. So everything expanded, but the division site did not. And it took us a while to figure out what it was, and in the end, we realized it's chitin. So there is a gene cluster for chitin in pombe. Nobody knew why it's there and how. We didn't go into the molecular details, but when we finally put also chitinase to digest the stupid ring, we basically ended up with a fully expanded yeast cell. And then combining it obviously with fluorescence is another issue, because, so if you just have normal fixed cells, so this is now a measure of fluorescence survival. In our first conditions, basically no fluorescence survived this shared protocol. And it took us a while, so we're not perfect yet, but we're in a way of that I would say it's useful or starting to be useful. We're currently with this protocol at a level that we retain roughly 50% of the fluorescence in a super resolved fashion. So this is now single molecule imaging with expansion microscopy. So this is kind of what I think all of you tackle more or less in your research questions, like similar samples like this, and we do it from microbiology. So that's our daily life. Okay, so study number 1. So this is, I think, when you would ask me what is kind of the limit in application, or what is currently possible when applying single molecule imaging to complement structural biological studies, like where you alternatively would do, I don't know, crystals or cryo-EM, then I would say this is kind of at the edge where we're currently at. So basically trying to figure out the stoichiometry and the composition of a full protein complex. And this is what we worked on quite a while. It took us, I think, in the end, 6 or 7 years in total to get this to the final stage. And this is the kinetochore complex as an example. And so for those of you who don't know it, so this is a yeast project. So it's a eukaryotic microbe. Pombe is very, very homologous to human cell division. So it's a model organism that is used for studying cell division and also studying cell division health, disease problems, etc. Right? So if you don't want to deal with human cells all the time because it has only 3 chromosomes, lots of genetic tools, division time of 2-3 hours. So there are many reasons why you maybe want to go with this simple eukaryote first. And the kinetochore links basically. So this is histones, nucleosomes, and DNA, like sketched very roughly. And this is the microtubule in cell division. And basically the kinetochore complex is this thing here in the middle that kind of links the centromeric DNA region of each chromosome with the microtubules that then in cell division takes the 2 daughter chromatids apart, right? And this is a complex that has roughly different 50 proteins being involved there, structurally both and regulatory. Subcomplexes of it, so it can be divided in the inner and the outer kinetochore half in different subcomplexes. Some of them have been nicely crystallized, others cannot be pulled down or cannot be reconstituted. And so the question is how does it look like, how does it work? And what we did is we basically, here the red stars, and I will come to that in the labeling slide a bit more closely. These are all the positions we basically put different fluorophores to map the kinetochore. Wait, wrong pointer, sorry. And so what we did is we created a big 3-colored library, so a genetic, fully integrated library. So we're working not with transient plasmids, etc. So it's genetic replacements, where we have 3 markers. One is a reference marker at the histone side, so it's SAMP1. It's the first protein down here marked in blue that sits directly on the DNA level in the nucleosomes. And the other reference we have is even not on this picture because it's far, far, far up. So basically, if you walk along these microtubules from the chromosomes to the center, from here, that's the spindle apparatus. And so we have a spindle protein that we mark the spindle plane. And then the 3rd color is one of those red stars. So that's the library. We basically have one protein of interest, so that basically makes the strain library, and we basically switch the protein of interest while keeping the references. And that's how it looks like. The spindle pole marker is diffraction limited because we don't need it that precisely. And then the 2 here are super resolved in a single molecule fashion. Again, wrong pointer, sorry. Okay, so maybe go one slide. So both of these tags here are genetic markers. So this is not an extrinsic staining. And the question was how, if we want to do that in a quantitative fashion, can we do dual colored genetic labeling in a quantitative fashion and read out in a complete stable cell line? And this took us a long, long while, and we tried a lot of things. We actually also tried extrinsic staining, for example, with Halo and SnapTex, etc., with nanobodies. But in the end, we ended up with a genetic strategy, and this is linked to something which we discovered in 2017, and in the end was super useful to solve this problem. It's a new mode of photoconversion. And these fluorescent proteins that photoconverge, I saw them in several talks, right? There is a family of the so-called green to red photoconverting antozone proteins that come from corals, and they all do the same thing. They're initially green, you shine in UV light, and then they convert to red. There are some examples like reverse versions where you can actually switch back. So we saw also some reverse ones, but the irreversibly ones, the original ones, all have here a backbone cleavage in the chromophore region that extends the chromophore and then shifts the fluorescence irreversibly from green to red. So these are all the names you might know: Dendra, Eos, Dronpa, Maple, KIK-GR, they're all from the same family. And depending on whom you compare with whom, it's above 70% sequence similarity. And what we found out also with other groups is the following, that you can actually switch some of them, not all of them, but some of them, not by UV light only, but you can basically shine in blue light, which they absorb in their green native state. That brings them in a short-lived dark state, which is still debated mechanistically what it is. It's most likely linked to the triplet state, but not the ground level of the triplet state. And from there, it has a ER absorption range that actually, while absorbing ER light, brings it, and this is now again for certain, into the same final red photo-converted state as if you would absorb UV at the first plate. It's not two-photon. It's really like via an intermediate state, and it's like living in this intermediate state, which is triplet state related. You can try to measure the lifetime. It's in the millisecond range. Okay, so that's cool. And so I will sum up what we now meanwhile know. I will not go into the mechanics of prime conversion, but I will tell you why it's good for and how you can use it. First of all, the proteins. What you can see here is a short sequence of the amino acid sequence here, which involves here the chromophore, so the HYG for the chromophore, obviously all the same for all these different proteins. And I told you that some of them could do prime conversion and others couldn't. And what we figured very quickly and then could also demonstrate is that it's basically to one amino acid only here, which interacts with the chromophore in one cases, and in other cases it's flipped away and interacts with another site charge on the side of the barrel. And depending on how this amino acid either influences the chromophore or is flipped away, this will influence your prime conversion ability. And if you take all these fluorescent proteins that have not a threonine, which you need for prime conversion, but an alanine or an valine, and just exchange this single amino acid, you basically make them all prime convertible. What is cool about this is that it reduces phototoxicity of single molecule imaging. So the UV photoconversion is obviously annoying if you want to do long-term live cell imaging. So what happens to microbes is that they die. They sometimes even explode on your slide if you do that too long and with too long power. The least drastic cell death is that they just stop growing. So they arrest and will not divide anymore. They also don't, so sometimes they only arrest, and then if you stop imaging and let them recover, then they will slowly start to maybe eventually grow again. But a lot of them also basically die and never ever will grow again. So this is kind of the typical phototoxicity which everyone deals with that does imaging with higher laser intensity or, for example, with damaging wavelengths. Prime conversion uses much lower conversion power, so the 488 and 730 you need are A, less damaging, and B, much lower in intensity. And so this is really, really nice because it reduces phototoxicity. And the third thing is we now had a tool for our kinetochore and other projects because we can combine it with another class of fluorescent proteins, which are DART, like PEMCHERry, photoactivatable fluorescent proteins. And I have to say it's still photoactivatable MCHERry, because so far nobody has managed to make a photoactivatable M-Scarlet or so. So that's on my wish list, but I know it's tricky, and so far it just doesn't exist, right? So PEMCHERry has its flaws, but it has one advantage. It really doesn't care about blue light at all. So it only, in its preform, cares about, here's the absorption spectrum, about the 4 100 nanometer light you shine in to convert it. So you can really shine as much as you want. We really stress tested this green light, infrared light, even the readout light here, it's the 561 laser. You can shine as much as you want. The PEM cherry, which is already there because it's in the sample, it's genetically there, right? It really doesn't care. Up to the moment you convert it and read it out. And that makes it a really, really cool multicolor strategy, which is quantitative and which we can count with. Yeah, and that's the magic behind this Kinetico story. So what we do is we first take the diffraction limited protein here, and we decided to make a flaw of M scarlet Pro. So M scarlet is a super bright red fluorescent protein. I think many of you will know it and use it, but it has one flaw, it bleaches fast. In our case, it's an advantage because at the beginning of every cell imaging, we just want to know, is it the correct cell cycle stage? Are we focusing on the correct mitotic plane? Basically, we're not angled, and we can also measure kind of the positions of the spindle poles. So that's what we use this reference marker for. And then we don't care that it's bleached, it's actually good, because then we can use the same red readout channel after this one is bleached away to first look at the protein of interest with the prime convertible protein, and then B, add the centromeric reference with PEM cherry. And in these both readouts, we're super resolved and we're quantitative. So that's basically what we do. I just set this reference marker, both super resolutions. It's in vivo, so we get distances and quantitative protein counts in vivo. And that's the image which we get thousands of times. We imaged lots of cells. We made a whole triple color library. So basically by swapping in the different proteins of interest, they're all tested for biology. So in our case, for the POMB specifically, you can destabilize cell division by throwing drugs that alter the polymerization of microtubules. So you can stress test that. That's what we tested by so-called spot tests, growth tests, if they still grow. And we also tested them for different temperature stress. And all except SEMP3, this one protein are healthy and behave like the wild type. So there is no, problems with those genetic tags. SEMP3, we later found out we were kind of stupid or didn't think too much, because in this case, the N terminus of SEMP1, like the blue marker, and the C terminus of SEMP3, the orange one, and these are the two termini we tag with the fluorescent protein. They're basically directly next to each other, and that was a steric hindrance, right? So that's why this protein didn't work in this strategy. Yeah, so strain library, spot test, flow cytometry for phenotype, cultural growth like shapes, to make sure those things are happy and alive and normal. Then a lot of imaging. So a lot of our business is not only getting the thing to work in an application, but then also to use it. So we have, I will not show you all 22, but in total we have 22 three triple color strains, which we all cell cycle synchronize to enhance the mitotic stage. When you look at a full cell cycle, I told you it's 2 to 3 hours, but roughly only 10 minutes of that are in mitosis. So you really want to, focus on that cell cycle stage. We meanwhile also do it in smart microscopy, just not synchronizing them before, but basically just picking the ones that are in the right stage. That also obviously works with an automated microscope. We do a lot of repetitions of imaging. So we end up with about 1,000 to 3,000 image cells per triple color strain. Not all of them are in the correct mitotic phase. This study is now extended to not only mitosis, but also covering all the other cell cycle stages. So you have your imaging data. You have to post processes to get the localization table and first super resolved images. And then a lot of things have to be done. And we also saw this in the talks, like drift correction, channel overlay, and annotations. So when you think about this, we have up to 6 clusters because it's 6 sister chromatids from 3 chromosomes. And we obviously have to cluster the clusters. We have to basically say which belong to which kinetochore. We have to identify the spindles. All of this has to be classified. And a lot of this is done in an automated fashion, but we realized that quite some of this is so sensitive and so difficile that so far, at least we didn't find a method that does it as good as humans would do classification. So I think here we end up with the same problem cryo-EM has, right? Very often for a lot of things, the human classifying something is still better than every automated method. Not for all of them, but that's why we have a mixture of manual and automated analytics. What comes out of this? I want to show you two things. One is the stoichiometry, like how are these proteins in the kinetochore, and the other thing is the distances in between these kinetochores. So how is the kinetochore built up? For stoichiometry, the readout with fluorescent proteins, therefore that they switch irreversible, is kind of quantitative per se. You can do quantitative readouts. Nevertheless, we used also counting standards to back up that our imaging technique basically is correct. And we use FTNA, so that's a molecule like a ferritin from E. coli that forms a 24-mer. And what is nice about it is that it basically first is a monomer, which forms a dimer. 3 dimers together form, 4 dimers together for an 8-mer, and 3 8-mers form the 24-mer. So the status before is an 8-mer. So you have a really quite nice contrast between finished 24-mers and 8-mers. So that's also easy to pre-detect and pre-sort. And so we put them into yeast and image them under the same conditions. And as you can see with how we image, a 24-mer will give this localization distribution, which in the mean is a bit lower than 24, but that's kind of our reference standard. Distances. Distances are a problem if they're as big as your localization error, right? So we can obviously nicely measure distances of, let's say, 100 nanometers. But when it comes to distances in the few nanometer range, like 5 to 10, maybe even 20, then depending on how good your imaging is, and fluorescent proteins have one flaw, they are not the brightest, fluorophores you could choose, then it's becoming problematic. And that's what I kind of indicated here. You have basically here the measurement error as a circle, and the real distances which you are interested in is in the same order of magnitude. And that's really hard to measure and disentangle then. And so here we thought a lot. We tried a lot of different measures. For a lot of reasons, we can't simply take all the data together and overlay it like you would do in, again, cryo-EM, tacking them all onto each other. What we came up in the end, it was a Bayesian approach. So we took all the measured positions and the errors within, and we basically now get a measurement triangle. But what we know is that in division, the whole thing is under tension, and along the z-axis, there's one preferred direction. There's one axis where there is tension and mechanical stress. So there is this one assumption in the Bayesian model, which is that there is this one preferred axis along which they align, and that's the direction of the distance we are actually interested in. And the error is in all dimensions, right? So the error is obviously isotropic. And with that, we were only left with the sampling problem. So if you have a Bayesian model, so writing down the model is okay. Finding the optimal answer and sampling the log likelihood of a Bayesian model and figure out now what is the minimum, so what is the most likely answer to your model is difficult, so you have to sample large likelihood space. And here we actually, I would like to advertise something. There's a probabilistic programming language called STAN, which I think we were the first to use it on single molecule data, and I can highly recommend it. It uses Monte Carlo Hamiltonian descent approaches. So the state-of-the-art approach is to deal with Bayesian log likelihoods and is super robust and super fast. And fast is here the important part to give you posterior distribution, like your answer, like the estimate of what you're interested in. And that's for us the distance between the proteins. So this is what we get. We get a lot of protein copy numbers. We got a lot of distances. We get even more, we get shapes of clusters, we get cell cycle stage, but I ignore that for a moment. I just focus on copy numbers and distances. And that's kind of the central graph of this study, which is, here's this model, more or less detailed, dependent on if there are structures known, or in this case, they're not so well known. On this axis, you have all the different proteins of the kinetochore lined up from the inner to the outer. On the y-axis, you have the distance. So the positions here are the distances of the posterior and the arrow of the posterior. And the color being the stoichiometry, so the protein copy number. And when we finally saw this, we were super happy because there are some obvious things. For example, here in this long proteins, which we have a structure of, we know that they are heterodimers. We know that the C termini are on the same side. We know that therefore that they're heterodimers, they should be in one-to-one stoichiometry, right? And that nicely lined up. So basically, you see this, and when we saw results like this, we were like, oof. But yeah, so there's a lot of things, and now I don't want to bore you with division biology, but there are a lot of things in here which were not known about the in vivo or in general about the kinetochore, because you simply could never measure it before in vivo in the cell itself, but only parts of it in subcomplexes. So where does this lead us? So what we did, we moved a lot, right? Yves basically said from Marburg to Pittsburgh to Bonn, and every time we took our setup, every time we took our strains, every time also people change. We have new students. And so here in Bonn, I think it was finally the time after 8 years to remeasure what we had measured first in the first two proteins, which we measured at the beginning were FT82 and NDC80. And so we were like, if we want to now continue the study, and that's what we do, we now, therefore that we have a healthy kinetochore map, we now go to the biologically interesting thing. So we can now screen for diseases that have to do with the kinetochore. We can deal with stress conditions that have to do with the kinetochore and understand this more. But what I wanted them to do first is to show that we still can measure the same thing. Yes, we can do. So these two proteins were now remeasured in Bonn by different people by a microscope that has crossed the ocean twice. And yes, we can still do it. The error is bigger because obviously we didn't measure it to the extreme statistics. We did it before, right? It's a proof of concept. So what is next? I don't show you that now because this is now really going into biology. But we are interested in disease models with the kinetochore. And that's kind of what we're currently working on, using all the established things for this. Okay. So I promised you dynamics. Ooh, this video looks interesting. Well, ignore all the color. The sensor doesn't have color. Adjust. So I basically ignored dynamics so far. And if you want to study single molecule dynamics, you have to add one step, and everyone I think here in the audience knows this. You basically not only localize your individual molecules, but you have to also connect them into a so-called trajectory. And that's, again, an algorithmic step, which in the first place sounds easy, but if you look at it more closely, it isn't. But if you do so if you manage to do so, then you get diffusive properties. You got also kind of, I call it diffusive maps here. So this is RNA polymerase in an E. coli cell. And you can see the red ones are the immobile ones. These are DNA bound, they're transcribing. And the mobile ones here are scanning the DNA content, which is already replicated in this case. So we have 2 copies of the chromosome and look for new sites. And you can also map this in probability distributions, etc. I want to show you one example. So what a lot of people do, and what is I would say to a certain extent also a lot of biological questions involve this, is kind of the difference between a bound state and a diffusive state, right? So a lot of tracking studies, which are biological, try to distinguish the molecules being bound to something from molecules that are diffusive. And I, again, want to show you where I think currently the limit is, which is distinguishing different diffusive species. So not immobile versus mobile, but mobile with different speeds and different stoichiometries. And here I want to introduce you to the bacterial injectosome. It's actually a common complex that a lot of pathogens use. A lot of stomach bug uses. Yersinia pestis for the Black Death uses it. So a lot of pathogens use this complex. Here you have the bacterial membrane. It builds up this static complex here, long needle. Up here, that's you. and then it basically infects you and shoots. Here they're called effectors. Most likely they are toxins. So these are the things that make you sick. They directly inject and shoot these toxins into your cells at the right place. So this is the type 3 secretion system of a lot of pathogens. And while especially the static thing here is nicely resolved, right? So you can see it's kind of conserved in between very, very different species. A lot of dynamic things, like how this actually works. This lower half here can detach. So that's the cytosolic part. It can detach from this green platform dock-on station and basically has the job to recruit those effectors plus chaperones and bring them to the needle. So all of this is hard to study and largely unknown. And that's basically what we studied. But before we go to these fast dynamics, there was even a slow dynamic, which was unclear, is basically the dynamics of this stator. And we found something very interesting here, which has to do with the infection pathway. So what happens is you swallow those. So these bacteria are usually in brachy, non-clean water, and you drink it. And then they pass your stomach at a low pH, and then they infect you here in the intestine. And if it's not a really harmful bug, but just an annoying one, it gives you diarrhea and stuff like this. Or this is kind of the bugs you usually have. But how is this needle orchestrated and regulated? It's not built from scratch. That would take too long. And we found something very interesting with a collaborator, which is it's regulated by partially breaking apart the static complex in the membrane. So it's very dynamic in the static thing, which nobody believed. And it took us quite a long while to convince a lot of people that this is actually happening. And this whole thing is pH sensitive. So they sense the pH. If the pH is low, they basically partially to 50%, this protein here detaches and disassembles. The complex stays in the membrane, so it's diffusive in the membrane to then reassemble when you go back to neutral pH. I spare you the controls and details and medical relevance of this study, but this is a very simple molecular mechanism which we could nicely show by tracking all these proteins individually with single particle tracking. Okay, but what about all this intracellular stuff? People didn't know exactly to which of these parts here, so these are the individual parts of this sorting platform, that's how it's called. It's a 6-fold symmetry. The front and the back leg I didn't draw, so it's 6, not 4. And nobody exactly knew to which one of them the effectors would be binding, because it's hard to purify them. Most of them have really hydrophobic scores, so it's basically impossible to do that ensemble outside of a cell. So what we did is we crossed out the whole system, and then we reintroduced only the individual pruophore. So STTQ, STTL, STTN, all of them individually. And then we co-expressed no effectors or with effectors. So there's no needle complex. It's just one of those proteins with the effector. And what you can see, not so nicely in these images, but what you can easily see in these graphs, is that for 2 of them, the diffusion slows down. And these are kind of the candidates which we know that they directly then most likely interact with the effector. What we did then is we have a strain that has no effectors of all. And then we have the wild type strain that basically here has the full complex. And here you can, in this system, tune it. Calcium inhibits secretion. EGTA chelates calcium signals away, so it starts secreting. And then you can also see that from here to here, you also have a self-regulation that once you're in the infected state, all the injectisomes here at the membrane, they get enhanced and they build clusters. So for each of them, they replicate, and next to it, you have more of them, right? So there are clusters of these needle complexes forming under secretion conditions. But these are not the ones we are interested in. We are interested in what happens here inside the cell. So ignore all the static things. We also did that. So all the data you now show is not interested in the static fraction. It's interested in the dynamic fraction. That's what you see. Without effectors, with effectors, but non-secreting, with effectors and secreting. And what we saw is immediately that there are lots of dynamics. So if you just look at the black curve, this is the weighted mean jump distance. So how far a single molecule goes in between camera frames, so basically jumps. It's kind of a the simplest measure you possibly could do on diffusion. So frame to frame distance, if you want to weight it over and for a mean. And you can see we have slower ones, we have fast ones, and here this is not Gaussian, so this is not one state. It has this very long non-resolved tails of a lot of different, what we think are different subcomplexes. And it's hard to interpret what we see here. But what we immediately kind of saw is that once you put the effectors, this whole black curve kind of shifts to the left and you get this red curve. And then if you put secretion on, you enhance this slow state. And we were puzzled for a long, long while. And I want to show you the final trick we did to basically, unfold this data. And this is this. From mass spec, it's kind of believed, so that's what the mass spec data says, that a single leg, so not a 6-fold symmetry, but a single leg is kind of a predominant cytosolic preform of the cytosolic sorting platform. And the stoichiometry, also here are debates about it, but the most commonly believed stoichiometry of this is 1 SCTK, 4 SCTQ, 2 Ls, and 1 yellow N. And we know the molecular masses of all of these. We know the stoichiometry. And now we know if you put an effector, how much it gets heavier, right? And we also know if we now take a single leg and kind of multiply it by 6, we get a full 6-fold symmetry. And yes, there's influences of charges, there's influences of molecular crowding, but we just assumed maybe the 1-fold and 6-fold symmetry are so similar enough to each other concerning charge distribution, etc., that if we just assume that if this here is a single leg and we see an apparent diffusion, a jump-to-jump distance from 270 nanometers, what would we think, assuming this diffusion and a molecular mass, which is 6 times more heavier, what should be then the diffusion or the jump-to-jump distance of the 6-fold complex or the complex with an bound effector, etc. And so we took this, put in this equation, fiddled around with the mass. That's the only parameter we changed, obviously. Put in the different stoichiometries and got to this diffusion coefficients. And that was for the first time that I believe that we actually can understand and predict. Well, actually, they moved. Interesting. So, okay, they're animated, so they should be actually more to the peaks. Anyways. But that's for the first time I believe that we actually can understand such complex, many different subcomplexes. So there are so many ways you can take these individual proteins and put them together in different pre-assembled forms that we can actually get a grip on what is happening within the cell. And now I spare you the details. We did a lot of more control measurements and backup measurements. We computed a lot of stoichiometries and different variations of subcomplexes and put that all together. And in the paper, if you're interested in how basically the needle recruits its toxins from the cytosol to effectively infect you, that's kind of the model we then finally came up with. Good. Final story. How much time do I have left, Yves? Like 5 minutes or so? I'm not sure exactly how late we started. Okay, perfect. So, boo me out from stage if you're bored. So, what is the magic behind this? So, for the static structural one, I told you Bayesian interference was important, like modern statistical machine learning methods like STAN. In this case, it was the tracking algorithm. So, if you have the correct tracks, so if you do this step here correctly, you can, I go back to my RNA-P simulation, or like not simulation, measurement here, because it's a simple system. You have transcribing ones, you have mobile ones, you have free ones. And you can just, by the diffusive properties, if the tracks are correct, then give quite quantitative numbers to this, right? You can basically say how much percentage of your RNA polymerase are actually currently working, or, on holidays, how fast that is, or transcription dependent on what it's transcribing, 20 to 100 seconds, etc. So you can measure quite a lot of different bio numbers. But yeah, basically tracking. This is the important step that opens up the whole field of getting as much information about your biology out of these tracks, like the dynamics, the spatial distributions, the state, the dwell times, if it's Brownian diffusion, all of this we are interested in. That depends on tracking. But tracking in microbes is challenging. So I show you here's patches of E. coli cells, not very fancy, just on an agarose pad. And if we do tracking, it looks like this. I'm not sure if you can track that by eyes, but it's very different from, you know, two-dimensional big mammalian membranes with one quantum dot or so. It's lots of molecules. They're a high density in a small volume. There are short tracks. This is a fluorescent protein. Again, it's dim, it's blinky, it's short before it bleaches. So it's not so easy to solve this tracking problem. And so for a long while, we have been working on this as well. And there are two things I want to show you. One is SWIFT, fast and probabilistic tracking algorithm. And the second one is TARDIS. That is basically, yeah, it's not giving you trajectory, but it's giving you population statistics about states. The SWIFT model, so the idea is easy. So if you, and that's what we usually do with the human eye. That's the reason why usually the human eye, and we are still so much better than the machines we train, because we kind of see a lot of information. So basically, the way we do decisions is not on how far did the particle jump. We also immediately look at the intensity. We immediately look at the surrounding. We look at the cell shape. We look, get a feeling for lifetime, the probability of something should merge or split. If there's noise around, how big the noise level is. All of this, we basically, if we do image per image hand tracking, that's kind of a human would always take into account. And now it's a basically simple arguing. If you can write a software that factors this all in, like the track lifetime, the probability of noise, the probability of motion change, the intensity, the blinkiness of the fluorophore. If you have all of these, the speed, obviously. So the most probable, like the most used parameter in tracking is kind of the distance, how far you can basically get, right? If you have all of these and then calculate for each possible connection you do between one spot to the other, each connection gets a probability value. And then you solve this very globally for the most probable solution given all the possible connections you want to do. Then you most likely have the best answer to your tracking problem, right? But it's problematic because to write that in a computationally efficient manner, there's tons of integrals you have to solve. That's the problem. But in the end, if you do so, and I don't want to bore you with the informatic details of SWIFT, but it's in the end a very efficient way to integrate a lot of integrals. Then you can do this. So this is the simulation of two diffusive states. And basically, if you sum them up again in mean jump distance, you see a slower one and a faster one. And if you analyze that with SWIFT, you see that's now the black line here. No, the black line is the ground truth. The histogram is what SWIFT gets. That's what SWIFT finds in the data in this simulation. And this is, meanwhile, so this benchmark is quite old. So there's meanwhile newer algorithms. But basically what not so informed algorithms will do, they will merge these two states. And that's of the death of analyzing your biology, because you suddenly mix states and different molecules you shouldn't be mixing with at higher density. And it also works on real samples. So this is CRISPR-Cas system targeted against itself. So it basically sits here on its own genome. And we can image this sample at low density and at higher density. So we image it at low molecular density so that you really, for most of the cases, 80% of the imaging frames don't have a localization at all. Only 10%, 5 to 10% have a localization in a frame, right? So this is really like, it's a simple tracking problem, because if there is a localization, then there is most likely no other. And then in the next frame, if there is still a spot, this is still the correct track. So you can go by kind of nearest neighbor analysis, or like connect to the next dot in the net frame. And then we image the same thing at 15 times higher density. And yes, you sometimes here have double spots, or like roughly 15%. But most of the time you still have 0 or 1 spot per cell. So it's not super incredibly high, but it's a bit more challenging. And if you do that with conventional tracking for the low sample, it works. and the results of SWIFT and conventional tracking kind of look the same. And now this is a different data set, that's why it looks different. And we didn't do it to craziness of statistics, but if you do kind of the same experiment at 15 times higher density, then you see what I told you before. Conventional tracking will mix those states together. So what does SWIFT take into account? Noise, measurement error, fluorescence lifetime is usually the particle lifetime and velocity, that there could be different types of movement, that there could be a motion change, that they could blink and reappear, and also that they could interact, and so trajectories could merge. Before you now get scared, usually your tracking problem is much, much simpler. So first of all, you put values into the program, but underneath each value there is a distribution. So basically it's not important to know if your movement is now 300 nanometers, 400 or 500 nanometers jump distance in between frames. It's maybe important to tell it if it's 30 or 3000, right? So to give it an order. It's important to tell it there's maybe 10% noise, but it's not important if it's in reality 5 or 15. It's to get a rule of thumb, because all these rule of thumb and distribution underlying, so they never go to zero, they have a peak, but they're all, not all of them are chi-square distributions, but many of them are basically, they never completely ignore something. It's just not very likely that a particle, which typically moves 300 nanometers, suddenly moves 1000, right? But it's not excluded, and that's kind of the trick. And that's why you typically don't need to know them very precisely, but you just have to have like good enough estimates for them, and then it already works. It can distinguish overlapping trajectories this way if one is slow and one is fast. It can correctly identify crossings. It can get rid of noise, and this is hard to see because it's so faint. But with blinking correction, it can connect fragmented trajectories. And it also, and this is also not to see, but here is a part of a trajectory that is immobile, and then it gets mobile, and it basically correctly identifies that this most likely is still the same particle. It just, in this case, went from a bound to a non-bound state. Yeah, so here's the example, again, on an E. coli cell. That's how it looks. So now the second thing, and I want to be quick about this, is if you think about this, you do the localizations, you do the tracking, you do the analysis. But you can directly skip the tracking maybe and go from localization data directly to analysis. This will exclude jump distances and squared displacements, because here you need the trajectory information, but it doesn't include analysis like simple jump distances, because this is our frame-to-frame linkages. And technically, you don't need trajectories for that. And that's the first thing you want to know. Population distribution analysis can be done without having the full trajectory. I want to do the disclaimer that there are also other approaches which are interesting to directly go from images to analysis and skipping the tracking, skipping the localization step, so to speak, and directly track images together, but I will not talk about this. So, and that's what we do in TARDIS. So we skip the tracking step. We just calculate all, super unbiased. Every localization we have has neighbors in the next frame, right? And so we do that for 1 frame, for 2 frames, for 3 frames. We connect a lot of wrong things, interparticle distance, but we also correct for the correct ones. And the trick is that there is a temporal distribution. So particles are on for a certain lifetime. And so at a certain point, you are left with the only interparticle distribution, and there's no real connection anymore. And that's the whole trick. Therefore, that Eve will kick me out of stage. I go quickly over this. You can do that for quite high densities where normal tracking algorithms, so these are the blind tracking algorithms. These are the a priori informed tracking algorithms like SWIFT. And here you can see that at high densities, if you don't give them the correct information, then it depends a bit on fine tuning. But if you give it correct priors, it still finds it. But TARDIS goes much higher. It goes to crazy scenarios like this, where you have 90% of true positives removed, 70% of noise, and it's still, here it starts to fail. That's a Jacquard index. So that's kind of when it starts to fail, but these are like craziness of scenarios. Nobody can image that or wants to image that. And what biologicalists are interested in multiple states. So what we do, we use SWIFT. It goes from localizations to segments to trajectories. Segments are if they're a motion type. So you want to basically not average over trajectories which have a motion switch between immobile and mobile, for example, right? So we do all these mean analyses on segments. And we use TARDIS directly from localizations and also back and even some more control data for more informed priors to go this step. So that's how we currently do our tracking. For everyone who does tracking, I can't go into details of that, but this is exactly the same data plotted in MSD, in a JD and MJD distribution. And you can see it changes by eye already what you see and how you interpret the data. So that's kind of the alarm. Think about how you interpret your tracking data. The event-based sensor. Here, a real quick 1 minute snapshot. 1 minute. Are you okay with 1 minute? Yeah? Okay, one looks annoyed, but the rest looks happy. Okay, 1 minute. That's the important sentence. It doesn't measure intensity. It measures changes in intensity. Yeah? So if you have this light on like this, it will not detect anything. So it will be pitch black. If I move, obviously if here is the pixel, you see my hand waving because here the intensity will change. So therefore that it only detects changes, it has very reduced data storage, which is cool for blinking data, right? We don't care about the 90% of dark background. We care about a few molecules that blink. And it makes it super fast because it doesn't have to constantly read out the full chip. It has a high dynamic range because it has a log threshold like the human eye. And that's what is cool about it. So this is how it looks, conventional image, event-based sensor. Gray is all the data which is not recorded. These are not changes. White are the positive, so increase of intensity. Black, decrease. So you get even directionality, you see the people here moving. It's used a lot in autonomous driving. What we were interested in together with Ignacio in Paris is, can it work for single molecule? And the short answer is, yes, it does. If you have a blinking molecule, you see it turning on, positive event, you see it turning off, negative event. And then we ran into a hen and the egg problem. We wanted to immediately characterize and benchmark that sensor. We wanted to know if it's better than EMCCD recordings, where the technology limits are and stuff. And then we realized very soon we cannot handle this event-based data. So it's tons of point cloud data, millions of events. You basically get a TXT file with events. And so we, in the end, ended up to first develop the software to handle the data. And now we're back to benchmarking. So this is Eve. And while developing Eve, we obviously handled the data quite a bit. So this is a two-colored image of an E. coli assay. That's RNA polymerase with a fluorescent protein tag. The pink is DNA paint. That's a three-dimensional boring microtubule in a living cell. The software has all the things you need also for normal software, like post-processing. But there are some things which are cool about event-based things, which I show you here. High density. You only see it turning on and off. The hole in the middle you don't see. You have the positive and negative event cluster that directly gives you an experimental localization precision. It directly, because of the fast readout speed, gives you the fluorescent on time, or maybe fluorescent lifetime. So this is DNA paint. That's why it's on time, because it's a binding and not a lifetime. It gives you in situ point spread fraction retrieval, in contrast to imaging data on normal chips, where this is really, really not trivial. And this is why I started it. The blue is the positive events. The orange one here, it's in the thresholds not symmetric. So focus on the blue one. The blue one is the positive event of a diffusive speed in this case. This is the recording of an EMCCD camera. So it gives you fast and super cool tracking. So that's also what we're currently working on most, dynamic Eve. Yeah. Candidate fitting, here's something puzzling. Really last slide. Log Gaussian is the best fit currently. That makes sense because it's a logarithmic intensity threshold. So if intensity is best fit by a Gaussian, obviously the events are best fit by a log Gaussian. But what I really don't understand is, why is this mean XY position so good? I don't know. I really don't know. It's surprising because it's much better than any other fit we did on the data so far, except for the log Gaussian. That's my group. Now you know why we have this image. And if you are interested in single molecule stuff, next year it's in Bonn. And if you're interested in more probes, there will be a next winter school by Manu, Dominique, me, and Claire in 2026. And the cool thing is this time we secured the funding so early and so successfully that I can say that it's fully funded, including not only the invited speakers, but all participants. So apply. Thank you.
You may have noticed I am not Aliénor Lahlou. She is ill currently, so I got to replace her, and I will try to do my best. So, compared to the other lab, we have a particularity. We are located in the northern part of Strasbourg, on the campus, on the CNRS campus in the north. So, basically, our main experience is related to synthesis of molecules, fluorescent molecules, more generally complexes, but we're interested in lots of things, going from molecules to clusters to very big nanoparticles, which can be used potentially for microscopy. This is one example, for example, with small organic molecules. I guess this should be the pointer somewhere. No pointers. Okay, let's try to understand what's happening. One of our collaboration, typically with Jackie Goetz and Alexandre Detap in Strasbourg, is to place some fluorescent dye. Here you can recognize the cyanine dye. No, you cannot recognize it if I can point it. So this, you recognize the cyanine dyes that we can put on a monomer part. And we are going to create polymer-like molecules with doxorubicin, which is a drug which can be encapsulated in the inner part of the polymer or at the outer part of the polymer, so that you will have a core cap polymer or an end cap polymer. And what we want to see is, can we break the bond, and is there a difference in the breaking of the bond to release the doxorubicin, the drug, as a function of the position of the drug in the polymer? So that's a collaboration with Jackie Goetz and Alexandre Detap, and typically that's what we observed in that case, particular case. There is no cytotoxicity of the polymer, and compared to the free drug, we can notice that, thank you, we can notice that depending on the position, there will be an action which is different in time, and the N is more rapid to react compared to the core loaded drug. This can be also observed by cytometry, by flux cytometry, and we also did some luminescence experiments, so basic microscopy staining experiments to reveal the presence of the polymer alone, for the polymer alone here, for untreated cells and for cells which have been treated with the polymer. So that's one example of organic molecules that we can do, or polymers that we can do. Another specialty of the team, and this is for more interesting maybe, is the fact that we know very well the coordination chemistry of lanthanides, and we are able to do lanthanide-based nanoparticles with very specific spectroscopic properties which might interest you. Basically, the properties are coming from the fact that lanthanides are 4F elements, and they have very long excited state lifetime. And when I say very long, for those of you working with a fluorescent probe with nanosecond lifetimes, here we are going to reach lifetimes up to milliseconds. That is very long excited state lifetime for europium or terbium, for example. Another advantage is that lanthanides are emitting due to FF transitions, which give you very narrow dyes compared to organic dyes, which are on broad scales in energy. And these will be, each lines will be typical of one element, that is one lanthanide. So you have a spectral signature for each lanthanide ion. The drawback is that the lanthanides are very poor absorption, so we have to play with a trick, which is called antenna effect, to put some organic antenna at the surface of the nanoparticles to make them bright. Okay? Basically, the synthesis is very simple. You take the lanthanide you want as their chloride salts. Here we use a matrix of lanthanide chloride, and we dope them with terbium or gadolinium if we want to have a mixed MRI fluorescent probes. Put them with ammonium fluoride in water in a microwave for 12 minutes, sorry, at 150 degrees. Centrifuge them, sonicate, and do 3 washing steps to obtain this kind of nanoparticles here. The applications of these nanoparticles, as I told you, here we have the bare nanoparticles. Alone, it is almost useless. What we did is develop all the syntheses around these organic molecules, which are in blue, what we call the antenna, which will collect the photon, give them to the nanoparticle, and the nanoparticle will emit. And we also develop the synthesis of labeling functions so that we can put proteins here or antibodies or whatever we want at the surface of the nanoparticles. Applications are typically in microscopy, as you can see there. This is we are targeting, in that case, EGFR receptor at the surface of the cells. And we can do also Z stacking experiment to see where the nanoparticles are inside the cells, at the surface of the cells, or out of the cells. If you're interested in this kind of nanoparticle, we have few examples, and these are now commercially available. You can even buy them by Polydetect, which is a startup company that we have developed aside from these nanoparticles. Another aspect of this kind of work that I didn't tell you before is that these nanoparticles are extremely resistant to photobleaching compared to organic compounds. There are a few orders of magnitude of resistance to the photobleaching with these nanoparticles, because in fact the energy that you will give to the ligand is not stored on the organic molecule but directly given to the metals, so that you don't break the bonds when you excite them. That's the reason why they resist to photobleaching. What we did with Andreas Reich and Nico Hildebrand is another aspect of this complex of europium, so that we developed this, we re-synthesized, they were published, they were known, these complexes, and we embedded them in organic polymers. We put them in organic polymers in an organic solvent, and by a nanoprecipitation technique which is developed by Andreas Reich, we can embed the complexes inside the nanoparticles. Then we can characterize the nanoparticles, as it has been done here by dynamic layer scattering or by TEM experiments, by microscopy. And finally, we can use them and measure the spectroscopic properties. Here you see a very specific narrow line of europium with a very thin line at 620 nanometers, which is in the red, so quite interesting. You can see that the nanoparticles compared to bare complexes are luminescent in water, still luminescent in water, which is not the case of the complex alone. And that's when embedded in the nanoparticles, we even protect more the lanthanide complexes so that their lifetime increases. Then you can use this for single particle tracking experiments, like that it has been done, or simple microscopy experiment with Z stacking also in that case. The interest here again is that you can do some labeling experiments or microscopy experiments for minutes and even hours without any important photobleaching. Another aspect is to combine different dyes in this kind of nanoparticles. This is still a collaboration with Andreas and Nico Hildebrand. And what we did here is that we mixed a cyanine dye with a rhenium dye. The interest of the rhenium dye is that its excited state lifetime is average. That is, depending on the environment, on the order of the microsecond, nanosecond to microsecond, typically. So we mix this in the presence of the polymer, do the nanoprecipitation experiment by mixing these organic mixtures in aqueous solution, obtain the nanoprecipitation, and centrifugate to isolate, or don't centrifugate, but we can then isolate the nanoparticles. Typically, if you look at the lifetime of the rhenium complex we embedded, it's some few hundreds of nanoseconds in solution. When you put it in the nanoparticles, we reach microsecond lifetime. And depending on the composition of each of the dye, you can see that we obtain luminescence, which is depending on the ratio of one dye compared to the other, with the possibility to make energy transfer from the rhenium complex to the cyanine. That is, from the rhenium complex, we can transfer the energy to cyanine so that we can have a dual two wavelengths imaging, for example. And also this has a strong impact on the luminescence lifetime of your cyanine dye. Okay, you can see that the nanosecond lifetime of cyanine can increase by up to more than 1 order of magnitude in this case, because the rhenium complex are long-lived, and this is transferred into a long-lived luminescence of the dye. This is also typically the kind of image we can obtain in that case with two-color images, depending on the wavelengths we are looking for. And last point I would like to add for this kind of nanoparticles is that we have been able to develop some molecular-based up-converting complexes. This is typically this kind of complexes that we have obtained by the mixing of very simple ligands with a mixture of two lanthanides, that is ytterbium and terbium. By doing so in this condition, it's very simple to obtain these clusters composed of 9 lanthanide atoms and which are dealing with each other. And typically when you take this kind of mixture with the this particular pair, ytterbium and terbium, you can observe the phenomenon of up-conversion that is exciting these complexes at 980 nanometers in the near infrared. You will obtain conversion of the energy from the two ytterbiums, combining the energy on a single terbium atom, and then emission in the visible, like this one, typically a green emission of the terbium atoms. The log-log plots confirm thus that it is a two-photon process, that is up conversion. And what we simply did is the same as before, that is take these nanoclusters, or these clusters, sorry, dilute them in organic solvents, acetonitrile or methanol, and do the nanoprecipitation technique to obtain nanoparticles containing the up-converting clusters. Okay. These are characterized by TEM. And we also see that these nanoparticles are also displaying up conversion in water in that case. That is, excitation at 980 on this level allows the combination of two ytterbium excited state into a single terbium excited state with emission in the grid. Okay. We can see also that there is a strong impact of water, nevertheless, even if it's at the nanoparticle level, there is still a strong impact of water, and especially the replacement of water by deuterated water strongly increases the signal. But we still have a signal in water, which is interesting for microscopy experiment. That's typically what we have obtained with Nico Hildebrand. That is leaf cell imaging of COS7 cells, where you can see the green spot arising from the up conversion signal. This is excited at 980 nanometer, and we record the emission in the green region. That are the spots that we observe. These are yet unpublished results, but I guess we should publish them soon. So it's apparently time, so I'm fully in time. Thank you here. I hope I've not been too long and not too boring. I would like to thank all the collaborators of the team, especially Aliénor Lahlou, who cannot be there today, but I hope I will have replaced her safely, and all people in the team. And finally, the students also, which are very important, not mentioned here, but the basis of our work. And I would like to thank you all for your kind attention.
Spoiler, ça. Okay, top. So thank you, and thank you for this opportunity to present our work. So today I will present high-resolution elastography. Okay, high-resolution elastography with optics, actually, which is one part of what we are doing in the team. So before we dive in elastography, a small word about our group. So I'm Amir Nahas, so I have the pleasure to be in charge of a small biomedical optic group of the IPP team of the eCube laboratory. So we are located in Civil Hospital of Strasbourg, and mainly we have 4 research axes. So fluorescence with Vincent, Raman spectroscopy with Amide, I don't know where she is, but she's here. Myself with coherent imaging techniques, and diffuse optics with Michele Diana. So the idea is to go from cell to organ. And today I will focus on elastography, which is part of the coherent imaging research axis. The initial motivation was really clinical, not too much biology, but we are more and more interested in biology and for small animal models, and in particular also for organoids. So here I have we have two. So as I told you, the really main initial motivation for all this work on elastography was the lack during surgeries for quantitative information to guide the surgeons. And basically they are mostly guided by the vision, actually their senses and experiences. So it was very subjective, and there is really a strong clinical need to have quantitative real-time information. And optics actually it's a good candidate. And also actually elastography. So why elastography? Because it's really it was used in medicine since the very beginning of medicine actually, through palpation. So the idea is when you have pathology, it will alter the stiffness of the tissue. So if you are able to detect the alteration of the stiffness, you should be able to detect a pathology. So through palpation is really a basic one. But more recently, you have a lot of elastography going through biomedical imaging with actually very good results, for example, here in ultrasound or in MRI. So for example, here you have 2 different lesions. On ultrasound imaging, you cannot do the difference. But if you have the stiffness, if you add, the stiffness map to ultrasound image, you're able to differentiate both. So you see that this one is more stiff. And actually for breast cancer, when it's stiffer, it's more dangerous. And here you have the same on MRI for brain cancer. And actually it's the inverse. When it's softer, it's more dangerous. So the idea is if we can do the same with optics, and all the advantages of optics that are non-contact, you can go from a resolution of the micrometer to a meter resolution without too much issue. So that's what we try to do, and also to answer a fundamental question, which is what stiffness at the micrometer mean, because it's not that easy, like for micrometer scale on tissue. So what is stiffness, and the link with the stiffness at this resolution to the stiffness at the higher resolution, which is a really complex question. So just for the basic idea of how elastography works, I will just focus on one technique that is, one approach for elastography. There is others, but this one is the most popular for biological application, for clinical application. So the approach is based on shear wave shear wave propagation. So the idea is when you have a homogeneous infinite medium, you have 2 types of acoustic waves that will propagate. Compressional wave, that are the wave actually you use to do ultrasound imaging or to speak. So for biological tissue, it's mostly water, so the speed is almost constant at 1.5 kilometers per second. And that's why actually you are able to do a tomography with ultrasound imaging, because it's almost constant, so you know the speed. You just need to measure time delay. And there is another type of wave, which is shear wave, that are polarized perpendicular to the propagation, which are much slower, so 1 to 10 meters per second in biological tissue, soft biological tissue, and has actually the very good interesting property, which is the speed of the wave is directly related to the shear modulus, which is actually quantitative value of the stiffness of your sample. So if you have, it's directly related, for example, Young modulus, if you are more familiar with that. So the idea is if you are able to measure the speed, local speed of your wave, you are able to measure quantitative value of stiffness within a biological tissue with some assumption. So for coherent imaging technique is great because we are very good at measuring displacement. So you are able to detect very low amplitude shear wave propagating. So for example here, it passed by the field of view, you will modulate your speckle, and so you have a very high contrast. And with actually any kind of coherent imaging, you are able to do this kind of detection of the shear wave. So a few years ago, that's what we tried at the beginning. So it's classical shear wave elastography. This is agarose test sample, optically homogeneous with a heterogeneity of mechanical properties. You can push here, look at this propagation of the wave, measure the local speed of the wave, and then you get a stiffness map. Actually, and you have here the Mickey shape stiffness map with a quantitative value of the speed, which is with some assumption directly related to the quantitative value of stiffness. So we were not totally happy with that because we need actually quite fast acquisition speed with small field of view, because 1 to 10 meters per second, it's slow, but still when you have a field of view of 1 millimeter, it starts to be quite fast for example, for 2D cameras. So for this kind of approach, you usually need a stroboscopic approach or a very, very fast camera, which are super expensive. So we were more and more interested in actually another approach, which is noise correlation. So you don't need to push and look at the shear wave propagation, the shear wave speed. You look actually at diffuse shear wave noise. So here, for example, you have, it's a simulation, but you have a noise. So it's really, actually it is a speckle, but for the mechanical wave. And when you look at this movie, the interesting part, so I will not go too much into detail of noise correlation method, but you already have the idea. The idea is you will not work on the speed, but you will work on the mean wavelengths. So here you see it's softer, so the mean wavelengths look smaller than here, which is actually stiffer, so the wavelength is larger, because you have this relation that links the speed to the wavelength if you know the frequency, and we always have, let's say, linear thing, so the frequency don't change whatever the medium is. Voilà. So that's the basic idea. So noise correlation is a good estimator for wavelengths, local wavelengths. That's what. Here it's the theory, but I will not go to detail. Really, it's like the correlation function give access to the point spread function of the mechanical wave. So what does it mean? It's if here you have your noise field. You take one point here in time, you correlate it with all the other points in this line, and you reconstruct here the point spread function at this position. You can do, for example, here at this position. And for the optic people, you know that if you have the point spread function and you know the numerical aperture, you have access to the wavelengths. So that's the basic idea of shear wave, noise correlation based elastography. And there is other thing, and I just mentioned it. It's actually you are not using this exactly. You are just using the top here, and on the top you have all the near field information for your mechanical wave, which give you access to a very high super resolution over the stiffness property. So here, for example, you see the wavelength is like this, but the stiffness map will be a much higher resolution, okay? Because you have access to the near field of the wave. That's what we did, so I'm going very fast. So it was a digital holography, an easy setup, let's say, to a coherent imaging setup to measure. So you have the noise field. Here it was the same kind of system with the heterogeneity, very corporate with iCube inclusion, soft inclusion, and you can retrieve actually very good stiffness map with this method. And we realized actually that if you know the numerical aperture, you don't really know to have noise to apply this method. And actually the thing I showed you before for classical elastography, it was processed with exactly the same approach, the noise correlation inspired, let's say, approach. And actually it's super interesting because classic method cannot retrieve this map with this signal to noise ratio, because the movie is actually, the signal to noise ratio is not that good. But with this approach, it was possible, and the signal to noise ratio it's really good at the end. So that's the basic idea for elastography. We developed the method, and now it's quite robust. So we want to push it to higher resolution and in 3D. And a good candidate, so it was always with the T of like surgery. A good candidate for that is it's full-field optical coherence tomography. Why? Because it provides images really similar to, qualitatively similar to histopathology. The contrast is scattering. So, but still you can do some equivalence between full velocity image and histopathology image. One limitation is only ex vivo because you need very high stability to do a classical full field image. So that's a limitation we address because we want to do in vivo and also wave propagation, and it lack of specificity. That's why we want to try to add elastography, so we go very fast. 1 year ago, no, 2 years ago now, we proposed a new approach for full field OCT. It's single shot full field OCT. It's based on well, it's a small technical trick, but now we are able to do one shot image of full field OCTs. So it's one shot image at a given depth of the sample with a micrometer resolution in three dimension. So it's look like this. For example, here it was on ficus leaf. And the movie is like every frame, it's like deeper in the tissue. You can do it in vivo, so it works really nicely. So here it's on the figure of a PhD student. I don't know if it's really, and here it's more, let's say, it's a project with a preclinical study on a mouse. So it should be a movie, yes. So it works really nicely on in vivo imaging. Very more recently, so a few months ago, we start to do propagation of shear wave on that, and actually we get this kind of movie. So it's quite new because it's very high resolution. The resolution here is like a micrometer, and the frequency of the shear wave is quite high. So we can go from 100 of hertz to 100 of kilohertz. So this is quite unique, and we can do a lot with this because it's a dispersion curve. We can do a lot with that. Well, here is the first result. It's not that nice, but we try now on ex vivo sample. It seems to work nicely, and we also have the method that work in real time for elastography, which is quite new also. So we want to try this on organoids and zebrafish. Voilà. So just to finish, thank you, and also thank to all my colleagues at the APV. Yeah, thank you.
Okay, thank you.
We are researchers from the Pasteur Lab, which is in the chemistry department at UNS in Paris. So today we're going to introduce a protocol that we developed with the rest of these authors that are listed here. And this protocol allows for imaging fast luminescence events down to microsecond range, using standard low-cost cameras that can go, only to low frequencies, which can't go so high, and we use periodic light modulations in order to. That's how our protocol works. So I'll first give a little bit of an introduction to explain the rationale of why we dedicated some time to work on this protocol. So in our projects, we're working on trying to image the physiological state of photosynthetic organisms, such as, in my case, plants, and with Hélène, algae. So we want to recover things like the water stress of leaves, or maybe the O2 concentration distribution in leaves or in algae. And so we aim to recover or glean this information from a couple of different things. So one being chlorophyll fluorescence. So when light is applied to a leaf, you get a lot of fluorescence coming off. So we aim to use the dynamic response of this fluorescence, but also we aim to use luminescent nanosensors embedded into the leaf or the algae, such that we can recover information such as the concentration of different analytes, say oxygen or maybe pH. And so I'd mentioned about the dynamics. So we're interested in extracting the information based on the dynamic response of these. In the case of chlorophyll fluorescence, so the plant has really complex mechanisms, and you have responses which occur within the microsecond range, but also going way up to the hour range or even longer.
And so, okay, it's easy to access the slower kinetics here with a standard camera, but down to microsecond, there we it's difficult to do with low-cost technologies. So it's one thing I should mention is that one of the goals of the project is to produce some low-cost device which will be affordable for a broad range of end users. And so also in the case of the luminescent nanosensors, they have lifetimes in the microsecond range. And so with a standard camera, it's they cannot be accessed. So we're looking at different ways that we could access these kinetics at low cost, or access these kinetics in any case. And we've seen lots of technologies which are super expensive, these film cameras with image intensifiers and all these things. But we wondered if in fact we could use something which is inherent in the photochemical behavior of our systems to be able to extract information about the kinetics at low speed. So we came up with two protocols called REOM and EOM, which allows us to access microsecond kinetics, millisecond range sampling. And so this can be done with a low-cost camera. So Hélène, she's going to describe like the general idea of how these protocols work, and then talk more specifically about the REOM protocol, given that we don't have too much time. So I'll pass over to Hélène. We'll give you this.
Thanks. So, in fact, maybe I need to lay down that. The two protocols rely on the behavior of your luminescent sample, where whatever luminescent sample you have, you can simplify its behavior down to a two-state model with a deactivated state and an activated state that will emit light. And the conversion between those two states, the concentration and so on, they are governed by some conversion rates. So in your illuminating condition between your two states, you will have an overall photo switching time, and this is what we want to measure. How do we do that? In fact, we are working with modulated light. For example, we saw the example with Aliénor where we were in time domain, so we moved to frequency domain. Because with modulated light, you will have your intensity of your light that changes with time. And the conversion rate of your luminescent sample, in fact, it is affected with the intensity. So your conversion rate will changes with time. So your light intensity changes with time, your conversion rate changes with time also, so the concentration of your two states also change. So this leads to a non-linear behavior that depends on the frequency, and also that can be predicted with stimulation because we know the conversion range, we know their dependence with intensity and so on. So in our group, this modulation has been done, and it appears that there are two observable, which mean two things that we can measure that will depend on the photo switching time. So something important to understand is that we will not do some demodulation. We talked about that earlier. We directly image something that has a fingerprint of the photo switching time. How do we do that? We image at several frequencies, for example, increasing frequency. We do a recording of our sample in those conditions, and then we compute what we call the REOM signal or the EOM signal. The REOM, in fact, it's simply the mean luminescence over time in an acquisition at a given frequency. And in fact, if we extract this parameter and we plot it against the frequency, it has a dependence toward the frequency. It seems like a cutoff function that we can fit, and we can extract the photo switching time. So this is a powerful tool. And if you looked a bit at our work, we validated on our beloved JAMPA2 protein. So here you have on the left, you have an image of HeLa nucleus cells stained with JAMPA2. In the middle, you have this observable REOM, so this mean luminescence over time for each frequency. And you see we have a cutoff function we can fit, and we can retrieve the lifetime. And we can retrieve this lifetime for each pixel. So at the end, we end up with a map of lifetime of our fluorescent protein JAMPA2. So this is, you see the lifetime, they are around the millisecond. So this is something you can do with your camera. Aliénor has shown it. So we wanted to see how down we could go. So I worked with metalloporphyrin dyes that are known to have excited lifetime around the microsecond, and that can be useful for oxygen sensing. And so I performed the REOM protocol on them, and I succeed in developing lifetime maps down to the microseconds. So this is an example of what we can do with REOM. And Jan will give you more applied example.
Thanks. Yes, so I mentioned at the start about this case of trying to extract information from leaves. So once all this was done by Hélène, so then I turned to leaves to see what we would get. In the case of leaves, the dynamics is very complex. Like in the time domain, if you apply light, you have like a tri-exponential response. So we're not really sure what we would see. So I implemented REOM on a different different system that I made to image over a larger area. So there I could image a whole small plant. And so first test I did, so I applied the REOM protocol to just a normal leaf untreated. And I had some kinetic response. But in this case, because it's very complex, doesn't really follow this two-state model, it was hard to find like a fitting function to extract some kind of time constant or whatever. So in this case, we leave it as this and call it a kinetic fingerprint. So we see that also the protocol can be relevant to extract rich kinetic information via these kinetic fingerprints. So I also tried this with, so I wanted to see if I could apply something to the leaf and have some different response. And so I applied an inhibitor of photosynthesis, applied the REOM protocol just so I could see, do we have a change in the kinetic fingerprint? And that's what we see down here in the bottom right. So last thing to mention, so because we could see this inhibitor, I wanted to see if I apply the inhibitor to the roots of a small plant, can I see the evolution or the uptake of this inhibitor into the plant? So what I'd seen is it can very selectively detect the presence of this inhibitor and its effects on the small plant. So just to wrap up there. So we'd seen, okay, this protocol could be used for, reversibly photoswitchable proteins for the photoswitching time. It's relevant for getting the lifetime of phosphorescent dyes. And also we can extract kinetic fingerprints in the case that our system doesn't follow a two-state model down to microsecond range. We've seen that it's easy to implement, so you just need to be able to modulate your LEDs and collect the fluorescence response using a normal camera. And then with some processing, you can extract the information you want. And then just as the last thing to mention, so we've submitted a paper with all of the details for REOM and the EOM protocol if you wish to use it. So yes, I'll finish there and be happy to take any questions.
Okay, thank you.
Thank you to allow me to talk about this subject concerning the development of a strategy based on the fluorescence lifetime in order to observe the tunneling nanotubes that are very delicate structure in living cell and by using red and near-infrared dye. So the tunneling nanotube or TNT are long intercellular communication mode, so described in non-mammal, mammalian, so bacteria or vegetable and mammalian cells. So the aim of this communication is to have a bridge and to transfer some element cell to cell. So the characterization of the TNT, it is the bridge and the non-contact in the substratum in vitro. And for the transfer, so element could be transferred cell to cell and by unidirectional or bidirectional way. And of course, it's a red light because it's less phototoxic, less photo-bleaching. And we need to have dye compatible with the cell alive to realize time-lapse, relatively long, and Z-stack. So Spirochrome and ProImaging propose some probes that are interesting in this case because the wavelength is red and near infrared. We have characterized the lifetime value and the monocomponent nature of this dye, and it's characterized very clearly the nucleus, the actin filament, and the mitochondria. So everything is very perfect to see the dynamic of TNT. And the lifetime mono-exponential allow to mix the dye together. We have other red dye allow to see very specific multi-component and realized lifetime imaging. And near red is very compatible with the STED imaging because the aim is to make a lifetime strategy by strategy of unmixing, but also with the STED nanoscopy. So for this, the cells are seed in a specific glass adapted for the microscopy, and the wavelengths of the dye could be very close spectrally, so that's why we have developed the lifetime based strategy. And we use j'ai les fenêtres partout. We use Leica Macrosystem Stellaris 8 to realize the image with the white laser, the Falcon module, film module, the environment, and the STED laser, the red STED laser. So from this equipment, we realize acquisition in confocal mode, but also in STED mode. And after that, we take the image to realize lifetime unmixing or lifetime imaging or lifetime denoding, so-called TOSTED. So we see the three possibilities. From confocal image, so this is a confocal image, so we have illuminate the two dye together and detect in one detector. So the confocal, it's only one channel. But when you observe the phasor plot, you have very clear 2 points corresponding to the mitochondria dye and the actin dye, and you can separate by phasor separation. After that, okay.
So they made me not the right slide.
Okay, I don't know why.
We too.
Okay, sorry. It's better? I think
yes,
it's
yes.
Okay,
yes.
sorry for this. So we can separate the different channel after phasor separation to have specific actin and specific mitochondria after separation, and after that you can clearly combine the two dye to have an overlay like this, and you can realize this by time lapse to see, for example, I don't know if you see the
we
pointer.
see.
Yes, clearly see the mitochondria at the basis of the TNT by time lapse. You can realize this, but add another channel to add another cooler. It is the case here. So you have the same thing, actin, mitochondria, but you add the DNA channel. And you can also have the 3 dye with only one illumination and one detection. And with the 3 spot corresponding to the 3 dye to have 3 channel, and after that, the combination of the 3 and the time lapse with here the fission of one bacteria, you can see, and after go to the TNT. And you can follow the mitochondria, for example, here at the basis of the TNT with a rearrangement, the big cargo of TNT and some TMTs that can go to the acceptor cell. And you can realize tracking, of course. If you want to improve the image, you can use the STED laser. So from confocal image, you improve a lot the resolution with the STED laser. And you can use the FLIM strategy to modify the image by toasted to increase the signal to noise ratio. And this is totally compatible with the living cell. So here in red, with only 2% of steady laser, we can realize time-lapse to see little wave inside the TNT, and also element that could be transport cell to cell. The mitochondria can be clearly seen with the steady laser. So it is the LBL from pro-imaging, and you see the mitochondria in one donor cell. The mitochondria can move bidirectionally, as you can see here, but also with one direction, linearly or not. And in the donor part, you can see the mitochondria that can go inside the cytoplasm. So you can follow instead imaging the mitochondria in the TNT by this strategy. So from steady image, you can improve by toasted, but you can use the specificity of the probe, so the mitotracker red have a phasor not mono-exponential but multi-exponential, so you can have some mitochondria in blue or red depending of the lifetime value. So this different color corresponding to the metabolic state of mitochondria, so it could be very interesting in biology. And to finish, you can use two detectors to see two different dye, use the modality of toasted to improve the resolution, and use in parallel the lifetime imaging to have the information about the environmental around the mitochondria, so the different type of mitochondria. And you can combine the informative image in lifetime imaging with super resolutive image to have at the end the combination of what you want. So this option is now developed by Leica with steady extend, but it's very interesting to have this possibility. And to finish, we have published some paper about the TNT and two other papers more specifically for the technique, and everything is described in this last paper. And we now are testing some homemade red probe, so with a collaboration with chemists of Rouen and Strasbourg, so it's in progress. And I leave my email if you want to write me. And I want to thank a colleague of Primasen team and collaborator from Caen, and thank you a lot for your attention.
So thank you for staying. Thank you, Loïc, for this two kind introduction. We started with the Lycée La Martinière because we just remarked that we were together at the same time in the lycée, the same lycée, but he was 2 years older than me. So I will thank also the organizer of this symposium for giving me the opportunity to present you the research we are doing in Lyon. I was trained as a chemist, as a pure organometallic chemist, but in fact I am a frustrated physicist, and I moved slowly from chemistry to spectroscopy. And in fact, I met a very old guy when I was a very young researcher that told me, don't forget about synthesis chemistry, because if you are doing synthesis chemistry, you will have the unique pleasure to measure your molecule you are made by yourself, and you are not dependent of the molecule made by others. And he was fully right. And so actually we are doing a lot of advanced spectroscopy, nonlinear optics, transient spectroscopy in the group. And we are very open also for collaboration. So I'm mainly working in 4 different topics. So the one for today is lanthanide complexes for two-photon bioimaging, but I'm also interested in pure photophysics, lanthanide photophysics. We have a very strange project dealing with lanthanide complex for protein crystallography. So if some of you are interested by making nice crystals, please try these small molecules that sometimes behave as a magic molecular glue, and that can help you a lot to achieve nice protein crystals for X-ray crystallography. And our last topic is the design of near-infrared dyes for nonlinear optics and bioimaging. So the keywords of my work are metal, colors, crystal, and spectroscopy. So today, in the context of the symposium, I will focus on fluorescence microscopy imaging. And as all of you know, you have three major issues in this topic. You have to get the highest resolution. And so all what I will show you is really low resolution compared to what I've seen these days, because we are just doing two-photon microscopy. We want to achieve the highest signal over noise ratio. So this is the job of chemists mainly, to design the best bioprobes. And we want to go in depth and typically work in this famous biological transparency window in the near infrared, which bring you a lot of issue in terms of signal over noise, signal, and instrumentation issue. So when you are doing microscopy, all of you know that you face with autofluorescence. And in most of the paper, in some paper, especially paper from chemists, not from microscopists, you see very nice mitochondria images with a lot of dyes, but you have absolutely no spectral, temporal characterization of the dye. And since now with the very good detectors we have, you can measure directly the autofluorescence from NADH. And so you can ask yourself, do I measure my compound or do I measure the simple autofluorescence of the media? So to tackle this drawback, it is necessary to make spectral microscopy and to measure lifetime and spectra inside the cells. And to do so, you have to face with the background signal, and your signal must, of course, be higher than the noise level. And this is characterized by a particular matrix called brightness, which is a product of the absorption and the quantum yield. Here I represent three different curves representing the same brightness. The brightness is the area of this curve. This is the level of noise. You see that the blue curves is below the level of noise. You have no chance to detect it. The red curves just reach the level of noise, so perhaps you will have a chance to detect this small part. But the dark curve is really super easy to detect. They have exactly the same brightness, but they have not the same width at half maximum. And so for microscopy, the sharpest bands, the better, because the signal for a given brightness will be much, much, much higher. And that's mainly the reason why lanthanide are attractive molecules for microscopy. Here I represent the comparison between fluorescein, a very classical dye, and the terbium complexes in the same range. You see that the terbium luminescence signal is very peaky, very sharp bands compared to the fluorescein. And if you want to draw the same area for the red and the green curves will reach the roof because of the very sharp transition. Okay, so the detection of lanthanide over a noisy signal is much, much, much easier than for an organic dyes. So that's what lanthanide guys said, of course, because you can make nice images with organic dyes. Okay, this is another version of the picture that Loïc showed you. This is a different lanthanides that may be interested for microscopy. So you see that the transitions are sharp, characteristic of each element. And you can mix all these elements together. And if you are able to measure a spectral of every lanthanide, you can have a multicolor imaging very easily due to the sharpness of the band. So these emissions go from the visible to the near infrared up to 1.5 micron in the case of erbium. And in this presentation, I will mainly focus on europium and ytterbium. Okay. This transition comes from the forbidden characters of the f orbital. So that means when the electron density is localized on the lanthanide didn't feel or feel less the surrounding. So it's an atomic-like transition, even if you are in a molecular complexes. This explains the sharp character of the transition. But these transitions are forbidden, symmetry forbidden. This is the Laporte rules. And so the consequences are double. They have a very weak absorption coefficient. And so it's very difficult to excite them. The epsilon value will be low, so the brightness will be ridiculous. To tackle this drawback, we will use what Loïc present you, the antenna effect. The antenna effect is what it's called, more rigorously, it's called the sensitization processes. That means you put an organic dyes close to the metal ion, you absorb the light in the organic dyes, and you transfer it to the metal ion. And generally, what is generally admitted is that you transfer it via a triplet state, an intermediate triplet state. So the classical Jablonski diagram is here. You excite it in the singlet, you make the intersystem crossing to the triplet, and then you transfer the energy to the metal ion that is emissive. And then you can take benefits from the huge absorption of the organic dyes and compete well with the poor absorption with the lanthanide if your transfer is very efficient. The second consequence of the forbidden character of the FF transition is complicated to get in, but it's complicated to emit. And so the lifetime will be very, very long, typically in the micro to millisecond range, depending on the different elements. And also the luminescence is also quenched by the presence of oscillator OH, NH, CH in the surrounding of the metal ion. This is exactly the same for organic molecule, in fact, and this is something that is really taken into account for lanthanide, but really poorly taken into account for organic dyes. That means if you made hydrogen bonds with an organic dyes emitting in the near infrared, you may lose a lot of emission due to the overlay between your emission and the overtone of the OH vibration. Okay, then you can take benefit of this very long lifetime to make what is called time-gated spectroscopy or microscopy. So very rapidly, you excite it here, all your organics lifetime and your noise, which is organic, basically organic luminescence, decay in a nanosecond range. And since your lanthanide decay in the millisecond range, you can just have a delay here and record in this spectral range. Of course, you lose intensity, but you decrease a lot the background. And so it's always easier to make microscopy over a dark background than to try to get over a threshold. So this technique is widely used to improve the signal over noise ratio and to monitor the biological event like drug discovery using a time-gated FRET experiment. Okay, lanthanide have only advantages then? No. Because if you want to make these sensitization processes, you need that your excited state or your triplet state stay higher than the lanthanide emitting state. And this gives you strong constraints concerning the excitation wavelengths. And most of the lanthanide are generally excited between 350 and 400 nanometer, especially for europium and terbium that are the most widely used lanthanide. And as you all know, this is really bad wavelengths for doing two-photon, for doing microscopy and especially in-depth microscopy imaging. And so one way to tackle this drawback is to go from 1-photon to 2-photon microscopy. That means you will excite the lanthanide with 2 photons of half energy, transfer and then you reach the same excited state level, and you can transfer the energy to the lanthanide, but with a 2-photon process instead of the 1-photon processes. And the 2-photon process are its intrinsic advantages for bioimaging. It is localized in the near infrared, so in the biological transparency window. It allows you to go in depth because of the reduced scattering. It reduces photo damage. It is intrinsic confocal. But this time you need to optimize not only the 1 photon brightness, but the 2 photon brightness. And you must use charge transfer chromophore to improve this famous 2 photon cross section called sigma, and to modify your complex accordingly. And so this is a summary of my CNRS research project I proposed 25 years ago, and I'm still working on that. Okay, so this is a benchmark of a lanthanide complex made in the 70s. You have 3 organic dyes that are stick around the lanthanides. And you have the energy transfer from the triplet of the pyridine to the lanthanide, and you can have very nice emission for almost all the lanthanide. But in this case, you have no charge transfer state. That means the 2 photon cross section of this molecule will be absolutely ridiculous. So what we propose to do is just to decorate this chromophore acting as ligand with pi-conjugated systems. Here we put 3. And we have good charge transfer from a donor group located here to the accepting group here, and then you can go to this stuff. And so this kind of molecule have quite large 2 photon absorption cross sections. So that has been done in the beginning of the 2000s. And we report, for example, this kind of molecule with 2 photon cross section ranging from 100 to 700 Goppert-Meyer, which are in the same order of magnitude than classical 2 photon cross section of organic dyes, because in fact, this is the organic dyes that control the 2 photon. The lanthanide has absolutely no rules in this case. But by doing so, we discover something else. We discover an alternative sensitization mechanism, because in this case, the triplet state is still present, but a transfer from the singlet state is also more probable than with other chromophore. And charge transfer transitions are strongly 1 photon allowed transitions. The epsilon coefficient of all the dye you are using is very high. And so the unexpected side effect is that by trying to improve 2 photon cross section, we improve very strongly 1 photon brightness. So the first compound that was successful was this analogous, where we just, from a synthetic point of view, it's a small just. We just put this hydrosolubilizing group, which is a pegylated unit. And so this molecule is perfectly soluble in water. And so we can make a spectroscopy, photophysics, 2 photon absorption in water. And the metrics are here. You have a quite good quantum yield, typical European spectra, a lifetime of 1 millisecond, a 2 photon cross section of 100 Goppert-Meyer. And I must just mention that thanks to this guy, Akosh, in our lab, we have now a 2 photon setup able to measure cross section that is very straightforward. And I think there is mainly 2 in France, 1 in Strasbourg and 1 in Lyon, perhaps 1 in Bordeaux, but there is less, in fact. Okay, then at that time, I didn't know anything about France-BioImaging. And when you are a chemist and you want to make bioimaging, you must find somebody that has cell, which is a very difficult task for a chemist. And the first guy that didn't say, oh no, lanthanide, I don't care, is this guy, Alexei Grishin. So I suppose most of you know him. He works in IAB in Grenoble, and he's a microscopist. And he said, okay, come on, we will try. And so we put this compound in living cell, nothing happened. So we have to find a trick I learned at that time. We have to fix the cell, so that means the cells are dead, but are looking like living cell. And the membrane are completely porous. So that means everything gets in, which is very convenient. And so the complex, in fact, stay and enter into the cell, are localized in this particular area. And we propose the first imaging of a lanthanide complex with a 2 photon excitation at 760 nanometers. and a detection in the red. But this complex has a problem because water is also a very good competitor for lanthanide coordination, and this complex is made of 3 ligands. And since the concentration of water in water is 56 molar, simply by mass action, you replace one of the ligand by water molecule. And so you have water molecule getting in the coordination sphere, and so the lifetime and the luminescent decrease. And so in the biological condition, generally we estimated that we have about 30% dissociation. So this first paper is absolutely a hold-up because we stain dead cell with a non-stable complex. But it was the first time. Okay, then we decided, okay, let's know what happened in the cell. So first we tried to stain living cells, and we tried to do some biological tricks, some membrane permeabilization, but without killing cells, typically using saponin, a small amount of saponin, you make small hole. The complex can get in, you can get this time-lapse experiment, so the cells are still living. They are reproducing, so normally it's something you do when you are living. And so this was, okay, we stain living cells. Then we want to go to microscopy, to spectral microscopy. But with a two-photon microscope and with lanthanide, you have a lot of problems. So for spectral mode, it's okay. You can use a classical spectral mode of your two-photon microscope, and you get this kind of profile, which looks quite similar to this one. But okay, the resolution is not fully adapted with the lanthanide complex because you have the resolution is fixed by the microscope, and our transitions are too small compared to the classical organic transition, but the profile looks quite okay. So we want to measure the lifetime inside the cells. And then we have a problem, because all the film techniques are made for organic dyes, so for nanosecond luminescence. Try to make film measurements for a millisecond emitters, it's impossible with the conventional techniques. And then you have another problem. The repetition rate of the titanium sapphire is 80 megahertz. So that means 1 pulse every 12.5 nanoseconds. And you have a lifetime of milliseconds. So 3 orders of magnitude slower than your repetition rate. So you cannot do film between 2 pulses. So we have to find a trick. And Alexei found that if you use the FRAP mode of the microscope, does that mean you just shoot 1 pulse? And if you use the temporal sampling of the CCD camera, so you are at the same point, you shoot 1 pulse, you have luminescence, and the camera is able to propose you a decay, a temporal decay of the luminescence. So the luminescence is so slow that the camera can detect it. So of course the signal is quite noisy. You made that 1,000 times, you convolute all this curve, and you get this very nice luminescence decay. The lifetime is 1.15 for a solid, and it's 1.10 when you measure it with a classical microscope. So we try that with our cells, and we measure a perfect mono-exponential lifetime, slightly noisy, but the lifetime is exactly the same, and Alexei calls this technique TS-LIM, temporal sampling lifetime imaging microscopy. So we are very happy. We contact Zeiss, say, wow, we have a fantastic new techniques for your microscope. You have nothing to change. You just measure the FRAP mode, and you can measure lifetime from 10 microsecond to millisecond. We must patent that and say, okay, the guy of Zeiss told us, nice, but there is nothing to sell, so nothing to patent, which is correct. In fact, every confocal microscope that has the FRAP mode can be used as a film imagers for long-lived emitters, but typically for ruthenium, for rhenium, for all these platinum, for all these microsecond long-lived emitters, this technique works. And Alexei write a routine, and so you can find it in this paper, but you can ask him the routine of that. So after 6 years, we go from, okay, a quite quick and dirty images to a complete spectral microscopy setup, and we can prove that the complex inside the cell is not destroyed, and the lifetime is identical, the spectral was the same, and so we have now all the tools in hand to continue the project. So the first thing we have to face is the instability of this complex. So this is a problem for chemists, and how chemists solve it. They replace 3 ligands by 1 macrocyclic ligand with 3 arms. And so we have just to decorate this arm, so it's only slide of synthesis, with our pi conjugated systems. It's not a so long task, but the purification of this microcyclic ligand is really complicated. Then you can build the complexes. You have exactly the same emission profile because you have the same symmetry. This is the metrics that are more or less the same. And this complex is in fact the brightest complex for 1-photon microscopy imaging because of this huge epsilon coefficient. So we never try to optimize the quantum yield, we try to improve the epsilon coefficient. This is the image we get. Always it's a fixed image because the complex is neutral, you don't want to get into the cell. And okay, it was fine. So based on this result, we said, okay, let's go now to the near-infrared, simply replacing the lanthanide by near-infrared emitting lanthanides. And so we took ytterbium, so we put a stronger donors because ytterbium has a lower energy accepting state, so we can put a stronger donors. And so we measure a quite good ytterbium complex, so 3 microseconds for an ytterbium gas in water is not so bad. The 2-photon cross-section is very high to the strong donors, so this is the classical spectra. You have the residual charge transfer band emission from the ligand because the transfer is not complete, and you have this emission at 1,000 nanometers from the ytterbium center. If we excite this compound with a 2-photon laser, the laser is here, we found exactly the same spectra with a residual emission of the ligand, and this band here in the near-infrared correspond to the ytterbium emission, and the slope is almost 2, which is very characteristic from a 2-photon process. Something that is not characteristic from a 2-photon process is that due to the huge pseudo-Stoch shift, these emissions lie at lower energy compared to the 2-photon excitation. For all organic dyes, when you excite at 2-photon, you get an anti-Stoch, formal anti-Stoch processes. That mean your emission is localized at higher energy, which is a principle of the 2-photon. Here, you have a band at lower energy. And this has consequence when you want to do microscopy. So we use always our fixed cell. We measure these images using the residual emission band of the ligand. But when we want to do the same experiments in this spectral range, in the near-infrared, nothing was possible because these stupid 2-photon microscopes, commercial 2-photon microscope, have filters to protect your eyes, and the filters cut the laser and everything below. And of course, if you want to open the apparatus, so generally the guy in charge of the platform is not very happy with that, and the microscope shut down. You say there, and you cannot replace this setup using the commercial 2-photon microscope. So I went to Sophie Bracelet in Marseille, which is a very good friend of mine. She worked in Mosaic in Institut Fresnel at Marseille, so pure physicist, specialist in nonlinear optics. And I told her, okay, build me a microscope that can measure near-infrared emission, but you made simply a filter that cuts the rest. So this is the simplest view of chemist of the job of physicist. She managed, but she took her 3 years, a complete PhD thesis, to make this alternative setup. And then with this setup, you can excite with a 2-photon excitation, but measure the energy at lower wavelengths. So you see that the spectra is not exactly the same than this one, okay? is because here we have just a CMOS detector, so that cut everything at 1050, and we are in the region where the detectors start to be very bad, in fact. So we just see what the detector can detect, and that was the shape of the signal is different. But it's a two-photon signal. And with this setup, we can make now two-photon images in a near-infrared to near-infrared configuration, exciting at 800 nanometers and detecting at 1000 nanometers of these fixed cells. We do that also with sick tissue models, which is slices of mouse brain. We injected this complex in the tail, and we detect the fluorescence in the near-infrared over a thickness of 65 microns, which is not so much, but it was the limit of our microscope. Okay, then we move to another lanthanide, samarium. So samarium is a very nice guy, absolutely neglected for bioimaging application because of its apparent low quantum yield of 1%. But samarium is a dual emitter. He has an emission band in the visible, and he has a tiny emission band in the near-infrared. So just for you, this is the quantum yield of 1% is measured before 750 nanopercent, and the ratio between this band and this band is 1/80. So that means there is not so much photon here, but there is some. And so we try to make two-photon imaging with the samarium complex, and we get surprisingly nice images of always fixed cell. And we can measure the lifetime and the spectra. We have these two bands here that are here. You see, the quantum yield is ridiculous, but you see clearly these two bands. So you are sure that it's samarium because of the sharp character of the bands. And even more surprisingly, when we measure the images in this spectral range, we get this image. And when we compare it with the ytterbium images recorded just before, okay, the image, the signal of a noise of the image or the contrast of the image is not very different. So that means samarium is as good emitters and compare quite well with ytterbium, but it has a much longer lifetime. So samarium is a very good candidate, in fact, for bioimaging process, especially because now in the visible we are very good detectors. We have picky signal, so we can take it. And in NIR, the signal here is in fact in the same order of magnitude of the one of ytterbium. Okay, we continue because I'm a lanthanide chemist with terbium, with dysprosium, but I will not bore you with that. And finally, we get a complete family of antenna that can either optimize the bioimaging of a given lanthanide, the energy transfer of a given lanthanide. So this one for europium and samarium, this one for terbium and dysprosium, and this one for ytterbium. Then we developed other antenna for sensing. This antenna is sensitive to oxygen. And we play a lot with this kind of antenna, stabilizing triplet state and making at the same time singlet oxygen generation and lanthanide luminescence. And that can be used, in fact, with gadolinium as singlet oxygen emitters. And this is the cells that are killed. So this represents more or less 10 years of work in the groups to make the proof of concept of bioimaging with two-photon stuff of everything, always with fixed cell. So it has absolutely no utility. So how can we do something useful? I know. We also generalize this family to many other lanthanide macrocycle, and it works absolutely in the same way for all other lanthanide macrocycle. And now in the literature, generally people, when they have a pyridine, they put an antenna like this just to improve the epsilon coefficient and gain in brightness. So if we want to do something useful for biology, we must keep the stability, the inertness, the brightness, so all what we get in the previous slide, but try to make, okay, why not small animal imaging, spontaneous internalization in cells, bioconjugation, theranostic, sensor, blah, blah, blah. You have a full list here. So let's try to show you some example for these three first topic. The first one, we just try to decorate these lanthanide complexes, so the same as before is a little bit represented in a strange way, by sugar at the end of the hydrosolubilizing groups. So we put glucose, mannose, and so many other. To improve hydrosolubility, but also because sugar are well known to be internalized in cancer cells or in cancer tumors. And so we have in mind, can these multiple sugar be a possibility to detect cancer tumors or cancer cells in sick tissue? So we try to stain living cells, and you see we remarkably stain the surrounding of the cell, but nothing enters. So this is the autofluorescence of the cell. So nothing gets into the cell. Okay. And then we go to Magalie Garibobo in Montpellier, who has a platform of small animal imaging working on zebrafish embryos, so very tiny stuff. So she injected our complexes in the tail of the zebrafish embryo. We wait a couple of days for the zebrafish to allow the zebrafish to grow, and then we made the imaging at 720 nanometers, getting the emission of europium in the red, and we get these nice images. So you see that the zebrafish is too big for two-photon microscopy, because two-photon you can just image a small area, and so you need 12 images to make a whole zebrafish imaging, but you have something that looks nice. But is it only europium everywhere? So to do so, we measure the spectral images of all these different images. So this is a zoom of the head, and you see that we have two colors, in fact. The orange colors is the europium emission. You see perfectly the europium spectra. And these red colors is, in fact, the autofluorescence of the zebrafish under two-photon excitation. So you have always to pay attention to autofluorescence, because sometimes the signal is as high as the signal you would like to detect. So then in the group of Magalie Garibobo, they are able to xenograft the zebrafish with the cancer cells that are growing, making a small tumors with their own vascular networks. And so in bright field, this is the image of this area. We can measure with a one-photon imaging the image of the xenographed tumors. Then we can image the two-photon emission of the europium, and if you merge everything, you have this image, and it's impossible to say if the europium is inside or not in the tumors. So we must go to the spectral images. The answer is not so clear for the moment, but apparently we have an amount of europium that is co-localized with the tumor. So actually it's impossible to say that it is enter in the cell or if it's just in the vascular network around the tumor, but that's something we are actually working on just to try to decipher if something happened or not. And we are playing with the nature of the sugar to see if we can modulate this signal. Of course, now we have our zebrafish, we have NIR emitters, so this is another complexes made in collaboration with Peter Komba in Germany. And so we try to image our zebrafish, but in the near infrared. So we mix Magalie and Sophie ability. And so in the near infrared, you can measure the signal in the visible or in the near infrared channel. So this is a control. You see that in the visible, you have this level of noise. That is, of course, increased when you have the complex, because you have the residual fluorescence of the ligand. But in the near infrared, you have no background. And here the image, of course, the contrast is not so good because it's a near infrared. But you see that you can detect the ytterbium emission in a NIR to NIR configuration. What about cell imaging? I tell you that the complex alone are never internalized in cells. It's because, in fact, the surface of the cell membrane is ionically charged, and my first complex was an anion. The second complex was a neutral complex. We have made some cationic complex, and cationic complex are spontaneously internalized. The other way is to use a cell-penetrating peptide to make the job and transfer the complex into the cell. And so we use this classical TAT peptide. It's a peptide that you can find in the HIV virus. And we grafted this peptide to the lanthanide complex. Once again, it's really complicated. The chemistry is really difficult because you have to desymmetrize the ligand. You have to make the grafting with that on beads. So this is done by Olivier Maury in Grenoble. And then you made this first complex. Here you have a cysteine. You make the dimer because the dimer is not to get in the cell, but to get out of the endosome and stain the whole cytoplasm. So this complex is quite pure. This is an HPLC view. They are luminescent. This is the toxicity. So if we go below 2.5 micromolar, it's okay. And so we can stain now the cell with this complex. And we were thinking, okay, we have done the most difficult thing by doing these huge molecules. Okay, apparently we make something. The cell looks stained. But if you zoom in, you start to be disappointed. And if you made the spectral mode, you see that you have this nice autofluorescence here. You have these big spots. And you have some luminescence inside and some luminescence outside. So what are these big spots? We try to wash, we try to make everything. We have still these aggregates, these big spots. And once again, the answer will be given by the spectral microscopy analysis. So we measure the lifetime and we measure the spectra of the spots and of the inside. And you must first know that the ratio between the two bands of the europium is very sensitive to the surrounding of the europium. It depends on the symmetry, it depends on everything. So you have a kind of internal probe of the symmetry of your europium ion. When you make the spectra, so in the autofluorescence, you see the autofluorescence. When you are on the green channel, you have the aggregate, and on the blue channel, you have the europium that is inside. You see immediately that the ratio between G = 1 and G = 2 is not the same inside and in the aggregate. 2.6 versus 8, and out and in solution it's more 10. So that means that inside europium is the same than in solution, but in the aggregate something strange drastically. When you measure the lifetime, the change is really important too, going from 0.7, 0.8, which is similar to what we have in solution, going down to 0.4. And this is a clear indication that the structures of the complex is strongly changes in these aggregates. So something happened, and we show that we can imagine that one arm is decoordinated and that something is getting here. And by doing some other experience, we show that there is an interaction with some proteins located in the surface. If you add proteins to this complex, you found things not exactly in the same range, but you see that the complex opens and that the lifetime decreases. So these spots are in fact at the surface of the cells and are due to interaction with proteins. Okay. I must say that the PhD student that makes this molecule was quite disappointed when she gets this result, but this is a proof that our spectral microscopy analysis is very powerful to analyze what happened inside. Hopefully, when we change the complex, when we go to the TACN macrocycle, to this Pickland microcycle with only one antenna, put exactly the same peptide, exactly the same staining method, this time we have very nice images without aggregation of this complex with the surface proteins. And more interestingly, we see that the staining of the cell is not homogeneous, and we see the two steps of the staining. First, this compound gets into the cell via macropinocytosis, that means get into the endosomes. And in this kind of images, you see some punctuation here, the same. And then it's been shown by Pelois, I think, yes, Pelois, a guy in Canada, that this particular structure enable endosomal escape. So that means the complex will get out of the endosome and stay the walled cells. And this is exactly what happens. So this is a kinetic process, getting in, staying into the endosome, and going into the complete organism. When we measure the spectra and the lifetime of this complex, it doesn't change between outside and inside. And so here, we finally succeed to make these cell imaging processes. Okay, I'm at the end. I hope I show you that lanthanide luminescence probe are really useful tool, especially when you are doing two-photon microscopy. So I call that the magic combo for the two-photon perspectives. It's just because Andrey is in the audience, and I would like to show you that there is not only organics. You can have bright, stable, functionalized lanthanide luminescence bioprobe from the visible to the near infrared. We made some instrumentation development with my physicist colleagues, so two-photon lifetime. We have also two-photon time-gated, I didn't show you. Two-photon NIR to NIR for cell and small animals. I hope I convince you of the interest of two-photon microscopy. And so the question is, what next? Actually, we are building some real bioprobe to act as sensors, with sensors of oxygen concentration. So this is a probe that luminescence and lifetime vary with the oxygen concentration. We have the same for oxidative stress. We try now to play with chirality, and we want to image the chirality inside the biological media. And for that, we have to build a two-photon circularly polarized luminescence microscope, which is another challenge. And we have new targets. We target sick tissue. We try to target tumors, but it's difficult. And very recently, we try to play with bacteria. Okay, this is the group today. So Annika is no more in the group because she moved from Lyon to Strasbourg because she's now a CNRS researcher in Loïc's group. This is all the people that have been involved during these last 20 years in Lyon. I would like to thank particularly Schoch and François, who are my favorite colleagues. So François is now in Bordeaux. This is my other colleagues in Lyon. Person in the PLAT team, I try to work with them actually, with the physics lab at ENSL. And all the co-workers, and especially the 3 microscopist guys that believed a long time ago in the potential of lanthanide, and without them, my career will not be the same. And I would like to thank you for your kind attention and for the invitation.
The idea is to debate about your needs. That's why there was a call to fill a small survey. So do we have the results?
We have the results. The questions. So the few questions were for the microscopist. We need a new probe. You look into a thyroid supplier catalog. Look into the deepest corner of your freezer. Call your chemist collaborator, or there was other. And the rest, we can maybe look at what they reply. Oh, but you made this.
Yeah, well, that's automatic.
Basically, yeah, most of you go online and spend money. Is that right?
And that's something which is funny, because typically, in the case of microscopists, 61% of you are going directly to see the catalogs. When we ask the same question to chemists, if they want to have their probes be used by biologists, then they look for someone to use it on a biological sample. That is, chemists are more confident in biologists than biologists are for chemists. That's maybe a shortcut, but not so far from this. Is there anybody who wants to intervene?
But there is no catalog of biologists.
That may be a
So
reason.
I think it's not fair comparison.
You're right. No, you're perfectly right. Also, we have to take into account that most of the microscopists are not using, not all of them are using probes, chemical probes. We have seen some techniques which do not need any probes, so those people are not concerned, really. Any other things to say about this point of view? Or any... Yeah, continue. But you know what? You can take my place.
In fact, no, it's
no,
not.
no, Andrey, that's not a joke.
Yeah, so yeah, in fact, the thing about approaching biologists, it's not that obvious because biologists, they have their precise questions and they have tools which are already well established. So when we come, we are kind of disturbing their standard way of working. And so this means you have to find a unique biologist who really look for a tool that does not exist. This is very rare. And so therefore, for instance, myself, I solved this problem. We build everything inside our own team. So we have our own biological facilities. Then it works very well. But finding a biologist who will do it for you, it has to be really a unique match. And it's not that easy. And again, there is no catalog for that. And so maybe those kind of networks, they are very useful to discover that there are problems that maybe we could address, but yeah, it's not that easy to find.
I guess you're touching an important point for biologists. They often have their focus on one point, but sometimes do not have the right tools to study them, or probably there should exist better tools. But is the discussion strong enough for them to be helped by chemists or vice versa? Maybe we have to improve this.
I think there's many chemists in the audience, and it's meeting like this one or like Mifobio, where the both community are sharing stuff that we can find collaborators, even if there's no catalogs. So they know us, we know them, and we can ask them something.
Lots of chemists, yeah. What about the biologists? We don't hear them.
Yes, hello. As a representative of biologists, yeah, I want to say that the probe in a catalog, usually it's well described. So you have a very clear protocol, how to handle, you have to fix all know that. So the protocol really elaborated. But for a new probe, already you have to contact someone, and often you don't have a good documentation, a good protocol.
Don't you think that we should try to improve? Thanks to
yeah, absolutely. Yes, absolutely.
So may we can come already at the latest question, which is how to improve the collaboration. Yes, of course. Okay.
It's not really a question, but it just we had the nice presentation of I don't remember your first name, Follet from Marseille. Even for the biologist, there is a strong barrier between I would say microscopist and flow cytometer, for example, where flow cytometer are using now a large panel of fluorescent dye on 14, 15, and biologists, we are stuck at 4 or 5. And I don't know why, in fact, because the dye exists more or less. And it's strange that even just few people are crossing this frontier. Coming to the chemist, yes, really, I think there is a need to for biologists to express the interest. I think there is big interest for live imaging now. Of course, fix is easier, and very simple things that what Andrey presented this morning. Just be able to label cell or the membrane with different color, and just the ideal for me as and what I'm working for. If I was able to only label the plasma membrane over days, it would be wonderful. I know it should be very difficult, but there's a kind of need that sometimes are not so on the paper. They are not so huge, but the solution is very, very complicated, likely for chemists.
Well, typically, I would say no. Because there are some possibilities, and typically the complex is that Olivier showed us are far less photobleaching than conventional organic dyes. But what we have seen is that for the moment is not able to label the membrane or something like this. But if there is a need, this need has to be argumented and told to chemists. If you tell Olivier that you're looking for the plasma membrane, I'm sure he will do efforts and discuss with you to target the plasma membrane and to show that his tags are able to be far more long-lived than conventional fluorophores. So typically the discussion has to be strengthened.
But there is biology also, they trap it.
Yeah. And then also problem.
Maybe an idea, we had the FBI image analysis challenges. Maybe we could have the equivalent with dyes and chemists, a kind of challenging, for example. Yes, anybody can try to work on a dyes for the plasma membrane for several days, and people try to work on that. But
so can you repeat the idea? I missed some
Just
point.
maybe to promote the collaboration between chemists and biologists, we could do a little bit like the challenges of France-BioImaging image analysis. So some people ask the needs, all the needs that we have in biologists, and the chemists maybe select one challenge and say, okay, maybe we can try to think about that and try to find solution.
Very interesting idea.
So I would like to add one point. Actually, the problem in this interaction, chemists and biologists, even if biologists express very clear need, frequently the solution is maybe not that interesting for a chemist, because when chemists try to find a solution for a relatively simple thing, it might be, in terms of chemistry, the solution is not that beautiful for publishing in a chemistry journal, because you do a conjugation of something with something, and technically for a chemistry it is not new. And so to have the best thing one can do is when there is a new chemistry and a new application in biology, which is almost impossible to realize. Frequently the solutions which are very beautiful in chemistry are quite useless for biology. This is unfortunate, because complex molecules are not very interestingly working in cell, because they start to behave themselves and start to decide by themselves what they do, because they are very complex molecules. In membrane probes, it's very well known. If you start making a complex membrane probe, it doesn't work. And so you need to really simplify the structure. And that's why there is a paradox. So chemists do chemistry, biologists biology, and the interaction doesn't work, because once you do it together, chemistry become less exciting and biology become less exciting. And that's somehow one of the problems. But then the people who find the best chemistry with the best question, those people win, yes. And then they get really new tools, etc. So that's really one problem I see when you try to design a probe, which is super useful. Frequently it is really relatively simple in terms of chemistry.
Yeah. If you see the other way, that chemists try to hear at the problem of biologists. That's already, you got a big problem for the biologists. You want to answer this. So if we are able to bring a brand new probe, which is also exciting for the chemist, then we can reach what you say, that is the gathering of both interest and big works, interesting works. So I don't see so much that it decreases the strength of both.
In fact, frequently we develop something nice and then we look what it is used for. And that's really common problem, because once you find, you develop something that somebody used once, it's very specialized. And so the opposite way, when we just say, oh, I could make with such mechanism, would be unique, nobody ever used that mechanism. You come out with a probe, it's kind of generic, but it has no specific application. And then we start looking for what could be the application of this. This is very common situation for a chemistry designer. But yeah.
Yeah. So I'm a biologist, and I want to say that the problem is not only if you have bright probes and whatever, if we can interact together. But the main problem is what a new probe will do to the cell, because when you work with living organisms, okay, we would like something brighter and nicer and etc. But we have to be sure also that it doesn't interfere with any biological question. So this is a long-term experiments to be sure that, okay, you see something, but is it what you are supposed to see, or it's your probe? So things that are, we know like 20 years now, we know what GFP is doing to the cells, we know some things. The new things that you prepare, they seem really nice, but do we know what they're doing to our cells? And do we have the time to? And so we have to have both ways motivation to go to this direction, because it's a long-term thing.
Old solution, which are not so new. For example, I prefer to design membrane probe based on cyanine because I know that cyanine will not kill the cell. And so I do not look for some exotic probes which I knew, which would be very beautiful chemistry, but I don't do it on purpose because I know that this probe will work and they work. And that's actually in line with what you say. However, I totally agree also with Loïc. We need to improve, and maybe we can improve drastically those derivatives of cyanines or in other probes going towards phototoxicity. This is ultra important problem because practically any fluorescent probe is phototoxic, and removing phototoxicity. Yeah, but interference is one thing, but phototoxicity is really a huge problem. It's even, I think it's more than anything. It's one of the worst things because when you do long-term experiments, you always kill the cell because of phototoxicity. But yeah, for sure. But that is even more difficult to predict.
Phototoxicity is important, but I guess that there are other points, such as with nanoparticles. There should be huge impact on the biological system too, not only the photosynthesis.
It's my turn. I would like to make a thing or parallelization between, let's say, okay, data analysis, image analysis, and chemistry. In image analysis, there is some researcher in image analysis that are doing new and complex algorithms, and there is also services because there is bioimage analysts that are at the service of the biology. In chemistry, it does not exist. And it would be interesting to think about, in the frame of France-BioImaging, to think about this. Is it possible to think about an open desk of chemistry in which we will be able to try to find a way to make a combination of simple chemistry or apply chemistry to biology or to the question of biologists. So okay, it's okay. You can answer that, yeah. Okay, thank you.
You know it exists. I think Ludovic Julien in Paris gets some funding from FBI for making a sort of platform of chemists working for a specific purpose. I don't know if. Yeah. It exists.
Well, it's very good to learn it. And I was even not aware of this. Ludovic Julien did this. Yeah, good. So it exists, but all the chemists does not know. It's only open to biologists, in fact.
So, as a chemist, we make dyes, and so sometimes we think we have a solution, but we look for the problem. And what may be interesting is we need maybe a platform for screening of all the dyes we made, because we usually characterize them in cuvette, and then after that we go to the imaging platform and we say maybe this one we make a selection after cuvette. So maybe let's try this one in cell imaging. But if we could extensively screen all the dyes we prepared on live cells,
yeah.
that would be maybe useful.
Yeah, but may I just a little point about this question? That's a very important point, because for the chemist, either you got a collaboration with a biologist who can do this and help you, but we can also do some efforts to learn cell culture and microscopy. For chemists, it should be possible, I guess.
So I don't know what I follow on the last suggestion, since we will have some a call of for transfer next year. So I'm asking for the chemist, and there are a few here. Do you think we could have, for example, the funding on the platform? I would say very basic imaging, where basically the chemist will be able to for 6 months or during the year to do a list or to send a list of compound, and that they want to have this compound to be tested for toxicity, for whatever, for signal in fixed and live cells. I think this is something that could be doable with a I would say a classical engineer, even a young a starter, a guy that is I would say not well trained or early in the career. So well trained, but not early in the career with no experience. I'm thinking, I'm looking for my words. So you think this would be an interest?
Very interested by this. Typically, actually, we have some collaboration which can enable us to do this with a biologist. But if one day I get angry with him and I won't have any more possibility to test this, and if there is a platform which can systematically test the cytotoxicity and do some few imaging of the probes, this would be very interesting, I guess, for the chemist. Yeah.
Do you will need specific, I would say, wavelengths and so on? So this needs to be defined.
Yeah, that's difficult. Yeah.
So this is just feedback about the comment that was given about characterizing all the probes and so on. So this is a message dedicated to chemists. If you need to characterize basic characterization of your probes, just have a look at the notes where you've got high content screening. This is the first step if you just want to know if it goes into the cell, where it goes into the cell, and so on. Then, if you want to have a deeper look, a deeper characterization about the probes, be aware that on the Bordeaux node, we have the material to, and we have some projects to analyze some probes dedicated to single molecule localization microscopy. So, and we have this kind of device as a collaboration between the R&D team and the facility. So basically, if you want to characterize your probes, we all have different levels of tools to do it.
Just to come back to that, so I take the chemist hat. So we are making a lot of molecules, and if we are waiting a biologist for making biology studies, a physicist for making spectroscopy, it will take few years. Okay, so if the chemist is interested, so I'm also making the cell culture, all the cell tests, toxicity, is it working in cells, is it co-localization studies, and even in the small animals. So it's very speed, and I think for students it's a good point because they gain a multidisciplinary approach. Okay, they will be less efficient in chemistry, but they will have learned to speak with physicists and biologists, and it's very, very rapid. But I don't have any biological question. Okay, I just see if the molecule is working, and I try to make a simple protocol for the next biologist. We try it for his biological question. So I think, don't take it bad, biologists, but you don't need to be biologists for making a cytotoxicity test or a confocal imaging. And now the microscopes are so good that you can even make a two-photon excitation spectra of your dyes inside the cell, just by playing with a computer. So the basic question can be assessed by chemists.
I
They want
agree.
to, and if they take the time.
I agree, Frédéric, but don't forget your background. This arises because you have been working on chemistry for two-fluorescent, two-photon probes for more than, I guess, 20 years. So that you have learned this, learned the culture cell, but basically what you have learned is chemistry and spectroscopy. Then you can learn to do the cells and then and so on and so forth, the microscopy. But not all chemical chemistry laboratory can do this. So that a platform, a platform doing this would be wonderful. Yeah, so we too. But how long have we been working? If a young scientist and a young chemist is interested in doing some fluorescent labels, can he do all this? No, for sure. He should be helped.
Yeah, I would like to come back to what we said in the beginning. Why do the biologists go to the catalog? Because the catalog exists. And I think we are lacking a database of fluorescent probes. And that's something that will be, that could be community-based, which means, I mean, the chemists could share the information on the probes they have, and this can be toxicity information, but it can be also physico-chemical parameters. You know, we have the FP base, you know, for the fluorescent proteins, and it was a game changer, you know, to really understand the huge variety of fluorescent protein that we have, and we don't have something equivalent for the organic probes. And I think maybe FBI or UBI can be a nice instrument, you know, to fund someone to build this database, and the chemists could contribute, and if we are looking for a new dye, maybe we could go to this database. I mean, that would be used by the world, I would say. What FP base is, yeah.
But I think we had this discussion in Yes. And in fact, the biggest, I remember we had this argument and counter-argument. The counter-argument against this database is that we are almost copying what private companies are doing, because when you commercialize a probe, this is what they do. They standardize everything and they put it in the catalog. So when you create this database, we're actually doing parallel work, but for freely available dyes, let's say. For proteins, it's different because these are plasmids, so you can reproduce them at home. This probe you can at home in your lab, you can resynthesize them in the cell, but organic dyes has to be commercialized, and so there is a paradox. Once they are commercialized, they go to catalogs of companies. Yeah. So, and these are products of our labs. So I don't know.
But for me, it's a way to publicize what you do in your lab.
Yeah.
And if your probe is there, I see this probe, I say, okay, and at the end, I see a reference, I see your publication, and contact Andrey, and I contact Andrey to get this
And
probe.
I think that is where actually the connection is lost, because once it goes to company, normally the connection with scientists is almost lost. And that's true, and this I agree, yeah. In any case, it would be a good idea to have this. I totally agree.
So maybe we need to have some catalog of chemical labs, like with different specialization in this domain, on this domain. I don't know, maybe I'm interested in probes for super resolution localization microscopy. Some other people are interested in some dyes to stain nucleus or just membrane, and chemists also have different specializations. So some use lanthanide dyes, upconversion nanoparticles, and they will not I cannot contact someone and ask to do completely different things that they do in the lab. But having maybe some list of chem labs and their specializations to contact them based on your needs, it will be also interesting.
But if such a database is well made, you have cross-reference, and you say, okay, I see this dye coming from Andrey's lab, and then I can click on Andrey, and I will see on the database if all the dyes that Andrey has been doing. And I mean, if it's well designed, and it's a lot of work, I agree with that. I think it would be super useful.
Can I return your remark? Marc, it is you're asking for chemists to prepare a catalog, but why don't we discuss of the need of biologists to be gathered in not a catalog, but no, that's it. Because then we can go in the different items, see this, what are the needs, because you have to identify your needs in terms of excitation, wavelengths, and so on and so forth. If you give this, we can try to see if our probes are working with this. So it should be in the two sense.
So I've got a tiny little question for chemists. When you're designing some new probes, new molecules, and so on, how do you store the information about the generation of new probes, etc.? Do you have some kind of an already existing database?
I don't think so. The existing database is, in fact, one of the answers of both questions. Typically, when we ask to biologists what they do, they look for collaborator, or they are going to the literature. For 40% of you are going to the literature. And for the chemist, that's the same. Okay. If you want to have information, it's around, it's a little bit more, 25% of the people are going to the literature. And that's the way to find the information. So maybe there is a gap between this need and this information that we have to fill.
But I meant as a lab. If you are designing a new probe, a new molecule, or whatever, do you have some ways to store a history of all the things you've been creating and so on? Some inventory?
Lab books,
okay, but because this could be the start of a database. You see what I mean?
Yeah,
instead of reinventing the wheel, maybe trying to see if you already have the information. And
I would say that the information is in the publications, but it's. Okay, but I don't think already, it has been gathered.
It's already the end product. It's not the intermediate product.
In fact, Fabrice is asking you if you have like secret probes that you're keeping.
Any chemist to answer? I don't want to say about my secrets. Frédéric?
Concerning the database, I think Luc Levis did a fantastic job on listing the probe he gets in the lab on the internet, and he's careful to always get some in stock. So you can write him, he gets the catalog on his site, and you write him and he sends it for free. It's not company owned, huh? It's really his work, and I think he gets 1 or 2 people working on this because he gets a lot of funding and he can afford that. Here in France, it's more difficult to get such a database and to be sure to get the molecule to send it to somebody. Because when the people making his PhD or his postdoc is leaving, he's giving you perhaps 100 milligrams of the stuff. And after, if you want some more, you have to make it again. So it's very time consuming.
Yeah.
The whole idea behind that, if there is a database that already exists, it means that some people have been thinking about the metadata to include and so on. And this could be a good template to start with.
Yeah, just to add, in fact, we worked before on an electronic lab book where all compounds we synthesized were there, but it is confidential. I don't know how could we share all what we are doing, because in this case it will be very difficult to compete with some countries which would look into it and do it much faster than us. So I think maybe it's different in biology where you want to share immediately everything you found, but in chemistry, okay. So, but in chemistry, certain things has to stay in the lab. But indeed, there are electronic books. For instance, we use for quite some time electronic book, and it's very easy to find all chemicals we synthesized. But we will share only those we want to share, and those are in articles. And then you have SciFinder that can find it, but there is no SciFinder for this is structure search database. But there is no equivalent of SciFinder in fluorescent probes. It doesn't exist.
If you think about fluorescence protein database, everything is also in the literature. What is nice in the fluorescence protein database, it's because it's very useful to work with. You don't have to read the literature, you know. So this is very important.
So I think, yeah, I'm really going back to what was said earlier. Maybe there is the testing by biologists. Maybe it's not so important, but the biological questions are very important. And I think as a biologist, and maybe you as a chemist, we are limited by a few things that basically prevent us to think out of the box, I would say. Maybe the limitation of the commercial microscope is the first thing. And the other thing is the thing that is not possible. And maybe we have to look in the trends, the new area where microscopy could go on. There are areas in cell biology which are very trendy these days, like metabolite dosage, like tension, like mechanical tension, like crowding, like phase separation, and all the very good cell biology is in this, and the DNA compaction and this kind of things. And so these are not always addressed by microscopy approaches. And I think because it's the closest to what I'm doing, but I think there are some of this where microscopy will go in. And so maybe if we think all together about this, we could find, I would say, real need and valuable need where basically if we could image some of this and quantify it, map this in the cell, it would be very nice.
It would be the reverse way, that is having the questions of biologists and coming back to potentially those making microscopy and those making potentially probes to answer the question of the biology, yes.
And there's also a point for concerning probes that you have probes that address that goes to a specific compartment by themselves, but there are others that you need to bioconjugate. And when we design probes, we don't always think about will they go directly to the target, or do we need to bioconjugate them? So in terms of design of the synthesis and things like that is different, because if we need to make them bioconjugable, gateable, so it's more complicated. So that's maybe not the first thing we think about when we design a dye. So
that's why I guess that the idea to have a platform dedicated to systematic investigation of the fluorescent property of fluorescent probes would be very interesting. The last for you.
Yeah, I exposed my quota already, but we talk about this opening the question, so what chemists can propose and what biologists can propose. It can be very nice. However, if you show it completely open, what is the guarantee that your idea doesn't disappear and is published after by people in Asia? Because they will collect all the best ideas that you put on the website. And because science is competition, huh? When we publish anything, it's a competition. We try to publish best idea and they don't accept the paper because the other one submits something similar. So I don't know how realistic it is to share all these ideas on the website. That's why usually it is private communication between chemists and biologists that work very well, and instead of putting everything on the website.
But the thing is that the biologist user doesn't need to know how you conjugated or built your molecule. We don't need, and then what would be shareable is just the functionality, the wavelengths and everything, but the precise atomic scale composition of the dye, we actually don't care as long as it works, and this can be shared. And as long as the toxicity is tested, but we don't need to know how it has been conjugated, compounded, composed. So some specificities could be shared, and the other part could be stored secretly in your lab book.
But tomorrow you will have 20 chemists contacting you from Asia saying, I have a solution. But FDA is a French group, so how to enhance excellence of the FBI and not excellence of other chemists outside of FBI.
So I think that this discussion has several layers. There is a layer that you're emphasizing, which is the need for the chemists and biologists that are doing research to work together to get new probes or to adapt existing probes. And this work can be valued by publication, patents, and so on. There is another level, because we are in infrastructure, proposing an access to users. And for our users to label their samples, they need probes. Some probes exist that are commercially available, and some are within the labs. So we would need a database of validated products that we could offer as part of the service of the infrastructure. So this is a second layer. And then there is even a third layer, which is the interaction between chemists and the facilities. Facilities where we have some devices to characterize your probes. We've got the knowledge to grow cells and so on, and to put your probes on our cells, on our systems, and give you some characterizations. So in this discussion, I think that we've been mixing the three aspects, and separating the three aspects to have proper discussion would clarify the panorama. Sorry. Come on, make some noise.
No, and to follow on what Fabrice said about the high throughput, I think that also in Marseille we are well equipped with a slides imager, like to do live imaging of tens of slides over days, and I think that the facility would be more than happy to have a collaboration with chemists to test the dyes that would be directly internally usable on higher end microscopes and like build a big project.
So you mean that we have to make an inventory of what the platforms should propose to chemists to test their products, so that biologists could see if the proposed products should be interesting for them or not. And this should be an open access, or with a minimum of information concerning the wavelengths, lifetimes, and quantum years.
And I think that even we might not even care if it works for us. I think that as a facility, we would just try it as long as there is some microscopy time instead of switching off the machine on a Friday. Then it could run over the weekend and be useful for someone.
This should be interesting. This come back to what we've said before. Yeah. And then, okay, so where is Bertrand Bercon? You will have some more job in the next coming days. Yes, and there was another.
C'est juste, yes, because it's okay. We are working with Pierre in the working room of expansion microscopy, and we have the same problem. We are trying to do a list of the different antibodies that work in expansion microscopy for the problems. It's very difficult. So we are trying to ask to the provider if they can maybe give them some antibody to test. And I think it would be very good if there is something with chemistry to apply for. I think it's the same for SMLM or expansion microscopy like that, because there is nothing about this.
An inventory.
Juste sur l'interaction entre chimistes et plateformes, je pense que c'est important de.
In English, please, Frédéric.
Pardon?
In English, please.
Oh, sorry. I start to sleep. So, sorry. Yeah, the interaction between chemists and platforms, I think it's very important for chemists not to send the sample, send 10 samples to the platform to try me that. I think it's very important for the chemist to send the student who prepare the molecules, because he feel it. You know what to do if it need a small amount of DMSO, no, it's better acetonitrile or water. So you get a feeling of the molecule, and I think high throughput is good. But I think artisan is good also. And the students, I think, have to go to the platform with the samples, even to discuss also what is done. I think that's one of the key points. The human point is also primordial.
There should be some teaching or some tutorial or things like this.
No, you have to go with the molecules.
Yes, but then you got a conflict, because on the one hand you have to have high throughput accessible to chemists.
I'm not so high throughput. So we are chemists. We are making 10, 20, 30 molecules for a series, and we can go for testing once a month, twice a month, to the platform for looking at the utilization and helping also the preparation of the molecule.
That's another word. There should be an equilibrium between this, where you fully learn to the student, and the possibility just to test them, because some of us.
But we get a lot of time this problem with the molecules, and it doesn't work. So there was some nanoparticles, some small precipitate, and sometimes they stop it. And if the student is around, he says, "Oh yeah, try with a little bit more DMSO, or try with 1 drop of acetonitrile," and it helps. And it's good for the student to see his molecules in works.
So if I try to gather together a certain number of things I heard. So we know that what is currently existing with the image analysis service works quite well. So you have a given problem in image analysis, and there is this FBIAS, which give you the opportunity to discuss with experts in the field. So you ask your questions. This question will be get by this FBIAS. They look at it, and the guy who is able to answer will be able to contact you and to consult with you for 1 hour. And if it is going further, then there is a connection and there is a real collaboration starting. So this tool exists, as you said before. You said that Ludovic Julia initiated something. I think this one we have to dig a little bit more and to have the same. So the communication will be inside FBI. It will not go on a website. So you send the question to the board of chemists. The chemists look to the question. They think that some of them could answer, have already something ready or close to be ready. He can then, okay, I am the guy, I will be able to reply. I be in contact with him, with the guy who asked a question for 1 hour. They discuss, and it's super important, because for even if you are educated biologist, you may not think to all the different problems. Is the wavelength suitable? Is it something which can easily go into the cell, and so on? There are many questions that chemists can have a good idea about, and you as a biologists you may ignore. And of course, in the other side, you have also some knowledge about your cells. You know that this type of cells have this specificity and so on. So I think if we try this, it would be a good start, because this will be, in any case, what helps is to have this communication between the two communities. So this is one of the ways to do the job. It's to show, okay, chemists have this product, this. So this can give ideas. Well, this is one way to communicate. It's to have this mixing of the two communities during these days. The other way, it's to have this kind of FBIAS-like chemistry board. And this is something which is of low cost, easy to implement. It depends just on the wish of the chemist to be involved in, to spend a little bit of their time. So from time to time, they will get some question and they reply if they're interested.
In fact, this point is important what you say. If it is inside communication, inside the community, it makes sense. If it is completely open, there is a problem of IP. Actually, we discuss, it's all ideology. But in reality, there is a question of IP, because when you develop a new tool, SAT will come and say, how you protect it? But we already communicated to the whole world about it.
That was the principle of the third question. Under which way should it be shared? Okay, should it be FBI work group? an open forum with frequently asked questions, a dedicated symposium, and so on. So it appears that FIQs should be the best possibility. Or according to the figures, 39% of the people have answered, this is the way we would like to work. That is almost 2% over 5 said we would like to have a forum for frequently asked questions. Okay. Or an open desk. That's more or less the same.
Simple to implement, and we are ready to start something, to contact people who might be interested to join, and let's try to see.
And we should ask people who want to participate, potentially ask them some keywords about their specificity.
Just the mail, just the contact. Because basically, if you can answer as a chemist, you show up. If you cannot answer, if you are polite, you send a mail that you cannot, or
you don't answer.
Okay.
You don't answer. So the idea is to say, okay, you will have 1 week to answer. So the mail will be disseminated, the biologist with some objective, or the microscopist with who has some wish, send the mail to the board of 10 chemists will get it, and if somebody inside has already something or knows how to do it or is able to address the question, perhaps it will be simple answer like, okay, but do you know that in the catalog there is already this product which is doing exactly what you want? Even that would be a great answer to somebody who may not have this expertise.
Maybe I would say something. It's not my duty, but is it possible to have a special grants every year to favorize interaction between chemists and biologists, to make dedicated grants on that?
The FBI tech transfer to target one for chemistry and biology interactions, and maybe master students or junior staff doing a few months in a lab. I don't know. That's how I got it.
We have to think about what would be the best formulation of the call, because since we will have very few calls, if you have 50 applications, there is no sense. But if we identify, maybe we can discuss a little exchange by mail, or you can see with Yves, if we identify a good way to propose this kind of things, we could do it. But the other proposition of Yves, I think, is very good, and I think I agree with Andrey and so on. We should be careful about any list or whatever or database that will be fully open to outside. It's dangerous.
In fact, this can be done with a single email list, which is the one of the 10. Generic one for the 10, or I don't know, the N chemists interested in, and that's it. So this will be a regular mail, no specific danger related to this singular mail.
But once more, I'm sorry, bloody hell, we should make some layers and precisely look at each individual focus, because a database is not something that is the big demon and so on. It all depends on what you put in. And if it's targeted at users with things that are commercially available, that have been published, because publication seems to be the hardest point in this discussion, if this is just a vector to disseminate, which is, I think, one aim within an infrastructure, to disseminate what the infrastructure is producing. And then for collaborations between biologists and chemists, when it is required, when nothing exists and when there is a will, then this is also one aim of the infrastructure. But of course, you won't put this into the database and make it publicly available, especially if you can do some valorization on it. So once more, I think that each time we are thinking about all these problems, which are not problems, because we are in an infrastructure and we can solve this, just let's think about in which focus we are and what we are talking about. The problematics in research are surely not the same as the one on the facility side. They are complementary and they feed one another. So let's have a look at, let's have in mind all those different focus, I think.
Is there any points that somebody would like to place into the discussion? Yeah. Okay, it seems that it's time. And I would like to thank you very much for the participation. Especially because, to be honest, at the beginning of the discussion, I was telling myself we would be 5 in the room and there would be no question. And finally, we took 15 minutes, so thank you very much.
Oh, we're 50, so that's pretty good. That's pretty good. I think we're...
So it is time to close this session and to close